So welcome everyone. As many of you know, for several years we've had the good fortune to host a series of symposia at the Graduate Center of the City University of New York, where we've had talks on a wide range of topics at the interface between physics and biology, very broadly defined, from problems at the level of single molecules all the way up to animal behavior and ecology and so on. And for the last several years, before the last couple of years before the pandemic, what we were able to do was also to welcome visiting students from up and down the Northeast Corridor and a few other institutions around the country that are part of an NSF network. This was wonderful fun. It enlivened things enormously to have kind of extra cohort of young people in the audience, interacting with each other, with the speakers and so on. Of course, this hasn't been possible for the last, well, 18 months or so. And as you all know, and in the meantime, many organizations have moved to a model of trying to have symposia from younger speakers to give voice to a broader range of people and ideas. And we decided that that would be a meaningful substitute for our tradition of bringing visiting students. So we reached out to the same people who used to recommend visiting students and said, could you recommend speakers? And perhaps we should have anticipated this, but we were really overwhelmed by the response. And so a whole committee of people, mostly students and postdocs themselves, made hard choices. And so we're delighted to have 9 really exciting talks lined up for today. As you can see, across a wide range of topics, the sort of full range of things that get covered normally over the course of an entire academic year. We're going to do it in one day. But what we also learned is that there are so many interesting people out there that we need to do this again. So we're thinking that this is going to become part of our regular rotation of events. Our center hosts seminars in Princeton, the symposia at the Graduate Center, and I think we'll now have these. So in keeping with the spirit of hearing from more people, I'm going to shut up and turn the chairship of the first session over to Kamesh, Krishnamurthy, and Trevor GrandPre, and they will tell you how things are going to work. I think the basic plan is, with short talks, interruption is a little difficult. We've asked everybody to imagine that they're talking for 15 to 20 minutes and there's a half-hour block. So there should be time for questions. And the chairs of each session will tell you how to manage, raising your hand, putting things in the chat or whatever. So Kamesh and Trevor, it's all yours.
Great, thanks, Bill. So I'm Kamesh, I'm one of the center fellows at the CPBF. And like Bill mentioned, just a few housekeeping rules. The talks are 30 minutes. We anticipate about 10 minutes of questions. And the idea is you post the questions in the chat, we'll monitor. If there's something particularly pressing, you can unmute and ask. And I guess I should let Trevor introduce himself. So he'll be looking at the chat and ask the speakers.
So hi, I'm Trevor, also CPBF fellow. And so I will be reading your questions to the speakers after their talk. Nice to meet you all virtually.
So great. So let's begin. And the first session is going to be entropy production and information flow in living systems. And we have Dominic Skinner, who's a PhD student from MIT, who's going to be talking about entropy production in living systems. So Dominic, go ahead.
Great, thank you. Let me just, here we go. You can see my slides, right? Okay, great. So thanks for having me. I'm very excited to be here today and to be talking with everyone. So as you said, my name is Dominic Skinner. I'm just finishing up my PhD now, and I've had the good fortune to work on a number of problems in biological physics. I spent some time thinking about multicellular structures such as bacterial biofilms and tissues and how we can use topology to understand them and compare them and so forth. I've also thought about biofilms more generally, how they grow, how you can control their formation and growth. Recently, I've been thinking about physics-informed model inference. So how you can use data to either constrain a class of models or to just learn something interesting and meaningful about your system. But today I'm going to be talking about entropy production. And hopefully, if there is time, I will explain where this cow comes from. But before I get going, I want to thank everyone that I've worked with, in particular for the things I'm going to be talking about today. This is in collaboration with my advisor, Yuhan, and a big thank you to Jasmine, Benjamin, and Alex for providing us with some experimental data to work with and apply our methods to. Okay, so when I learned thermodynamics as an undergraduate, what I was learning was a macro scale equilibrium thermodynamics. And as it was developed in the 19th century, the applications were engines, and you can describe an engine as doing some quasi-static cycle where everywhere on the cycle it's either in equilibrium or at least near equilibrium. And with this equation, with the framework, with the equations, the ideas, the concepts, and so on, you can start talking about efficiencies, and you can start talking about works and power and so on. And of course, you don't need me to tell you how useful and widespread thermodynamics is as a theory. But when we get down to the small scale, you can imagine plenty of little engines in biological systems. I mean, the classic example here is the bacteria flagella motor. You would like to treat these thermodynamically, but you cannot apply this equilibrium thermodynamics to these systems. Okay, so they are operating far from equilibrium. There's no concept of a Boltzmann distribution. There's no quasi-static states. And moreover, we're not necessarily averaging over 10 to the 23 particles. We might care about a single strand of DNA that we're pulling apart with optical tweezers. We might, in the bottom right here, we have what happens when you attach a bead onto the end of one of these bacteria flagella motors. And you can see the trajectory. I mean, it's tracing out a circle, but it's stochastic. Okay, and so these two ideas that the system is going to be far from equilibrium and trajectories are stochastic are key for these small scale systems. I mean, we can't use this theory of macro scale equilibrium thermodynamics. Now, fortunately for us, and quite remarkably, in the last 20 years in particular, the theory of stochastic thermodynamics has been developed, and this allows us to talk about these small scale out of equilibrium systems with the language of thermodynamics, all of our favorite things. We can talk about free energies and entropies and so on. And I don't have time to go into the full history, all of the technical details, and everything like that. This reference is a good place to start if you're interested in that sort of thing. But here's the idea. Okay, so maybe we only care about a single enzyme, and we care about the enzyme and how it's interacting with some cofactor molecule. Okay, not 10 to the 23 enzymes, just one. But it's still true that we don't care about all the velocities and positions of the water molecules in the bath. That's not important. Okay, the exact fluctuations around a particular state is also not important. The things you might care.
Just give him a second.
Hello,
Dominic, we just lost you for a couple of seconds there.
Yeah, sorry about that. So what I'm saying is that we don't need to know about all these specific internal configurations. We maybe only care about these mesoscale states. So I'm going to call these mesoscale states, and we combine all these different microstates into one mesoscale state. And if we do this properly, if we do this carefully, in particular, there's a time scale separation between the fast dynamics of the bath and fluctuations and the slower time scale of switching between states, switching between the open configurations, closed ones, so on. If we have that, we can define a thermodynamic theory on the dynamics on these mesostates. Okay, and what we're going to see is Markovian dynamics on this set of mesostates. Okay, why would we want to do this? What do we get from this? Well, as in the macro scale case, we can now start talking about the efficiency of a particular process, whether this is a molecular motor or something else. And we can also start to quantify some of the trade-offs, the thermodynamic trade-offs which systems have to make. So for example, with active sensors, there's a trade-off that you can do with, you can pay more ATPs. Basically, you can put in more energy and sense more, to sense better, sense more accurately. And for example, if you want to keep time in a biological clock, again, there's a trade-off between how well you can do that and the thermodynamic cost to how much free energy you're willing to put in. And there are also ideas of fundamental limits. There's some, thermodynamic restrictions on how well you can do something and how fast you can do something, and so on. So this is it's a very exciting theory, and it has a lot of applications, and there's a lot of reasons why you'd be interested in it. So how does this work in practice? So let's go back to the system that we had before and say it's a system on 4 states. And I'm going to say that you transition clockwise at a rate K plus and counterclockwise at a rate K minus. And so if K plus is bigger than K minus, this is a typical trajectory you might observe. You're typically going clockwise. You might take a counterclockwise step, and so on. Okay, so those are the dynamics that you observe. Now, there is this very remarkable result that what we can do is we can have an equation which on the left-hand side is a thermodynamic quantity, and on the right-hand side is a quantity we can compute from a trajectory. Okay, so the left-hand side is the entropy production rate. So what do I mean by that? Well, if we have a system like this one that's out of equilibrium, so if K plus is bigger than K minus, right, you play the video forward in time, it's going around clockwise. You play it reverse in time, it's going counterclockwise. That's violation of detail balance. The system is out of equilibrium. For the purposes of this talk, everything I'm going to talk about, everything I'm going to think about will be in a steady state, so a steady non-equilibrium state. It doesn't have to be that way. The theory can extend, but for today, everything's in a steady non-equilibrium state. So if you're in a steady non-equilibrium state, you're being kept away from equilibrium by various reservoirs, whether that's thermal or chemical. And so you're increasing the entropy of the universe as you maintain yourself out of equilibrium. And so the entropy production rate is the rate at which you're increasing the total entropy. Okay, so that's on the left-hand side. That's a thermodynamic quantity. And if we care about biological systems, these are typically isothermal. And so if we multiply that by a factor of T, temperature, what we'll get is the rate of free energy dissipation. So that's a really key quantity that we might like to compute. Now, the right-hand side of this equation tells you can compute that from a trajectory, from statistics which you can measure by just watching a trajectory. In particular, the ratio of forward and reverse probability. So you see a forward path, how likely are you to see a forward path versus how likely are you to see the time reverse counterclockwise? Okay, so it seems all we need to do is observe the system and compute this quantity, and then we'll know something thermodynamic and we'll know something really interesting. But here's the kind of central challenge in this. Even if the dynamics on these mesostates are well defined, and you can well define this thermodynamic theory, you only get to observe the system from a coarse-grained perspective. And so I'm going to imagine here, for whatever reason, states 2 and 4, we cannot distinguish in our experiments. We only see a single state B. Okay. So instead of seeing the full trajectory, we see this version, this coarse-grained trajectory. And we're left guessing as to what the true system is. We don't know how many internal states there will be. We don't know what the transition rates are. All we see is the coarse-grained dynamics. Now, there is this result, which is well-known and widely used and very useful, which just says that you can take this coarse-grained trajectory, you can plug it into the same formula that you had before. So you can look at the forward coarse grained, the reverse coarse grained. And that is now a lower bound on the entropy production rate. Okay, so you take your coarse grained trajectory, you apply the same formula, that's a lower bound. Okay. But in this case, in this particular system that we're looking at now, when we've done this coarse graining in this way, the observed trajectory is time symmetric. And so this bound is now trivial. So then the question naturally becomes, well, can we do any better? And so I want to think for a second about, when we think, can we do any better? I want to think about what would be optimal, what would be the best bound. And then from there, we're going to come down to something that is practical we can actually do. But bear with me when I explain like the idea, what is the best bound? Okay. So the idea is that we have the true system. I'm going to call it the true topology S. What do I mean by that? I just mean, the number of internal states, these mesoscale states, and the transition rates. Okay, that's what I'm going to call the true system. We don't get to see that. We get to see coarse-grained observation. So I'm going to say we get to measure this set of observables. And by this set, I just mean that any statistic which you can dream up that you can measure from watching dynamics on these coarse-grained states. Okay. Now, we have a set here in green, which I'm going to say is the set of systems which are indistinguishable from the true system. And what do I mean by indistinguishable? I mean that they agree on every single observable. So anything you can think to measure, the true system agrees with this other system. And so I, as the observer, only observing from a coarse grain perspective, I cannot tell them apart. I cannot know which is the true system. Okay. Now, therefore, the best bound that you could ever hope to get is you could say, well, I don't know which of these is the true system. So if I take the minimum entropy production rate out of everything in this indistinguishable set, that's a lower bound, and that's also the best lower bound that I can get. So minimize over this indistinguishable set. Okay, so that's the best bound. It's all well and good for me to say that. It's actually totally impractical. Okay, it's totally impractical to do that because if you are a Markovian system, you only have to measure transition rates, number of time I see I to J transitions, and then all higher order statistics you can compute from knowing those transition rates. So that's all you need to measure. If you take a Markovian system and you coarse grain it, the dynamics on those coarse grain states are not Markovian. They're not semi-Markovian. They're not second order Markovian. They're not anything like that. In fact, there's an infinite set of things which you could measure. And some of these, there's no way to get it to a finite set of statistics. Okay, so it's obviously impractical to measure an infinite set of statistics. So the idea that we're going to do is then we're going to say, well, let's relax this a little bit. Let's take a slightly larger set. And because we're taking a slightly larger set, it's a slightly worse bound. But now let's just take a set where everything matches on two statistics. So let's say the number of IJ transitions we see, so how often it jumps from one state to another, and also the number of IJK transitions we see, so how often it does this IJK transition jumps. Okay, this is practical. So we can now measure this set, and we also go one step further and say, let's just measure the IJ transitions, slightly bigger set, slightly worse bound, but again, it's practical. We can actually do this, we can measure it. Okay, for this set, which I'm calling O1, so we just measure the I to J transitions. What we can show is that you can take any system you want, you can combine these meso states in such a way that you reduce the entropy production rate and you preserve the O1 statistics. And so you end up with a system with one internal state in every macro state. And the bound that you get from this is previously known, and it amounts to saying, pretend the system is Markovian, pretend your observed dynamics are Markovian, and you'll get a bound. And if you go one further, so you take the number of I to J transitions and the IJK transitions, I'm not going to go into the details, the technical details of this, but the idea is what we can show is that you can take any initial system, any number of transition rates, and you can do some transformations to this in such a way that you lower the entropy production rate, you preserve these statistics, and you eliminate internal connections within a macro state, within a coarse-grained state, sorry. And so if you do this procedure repeatedly, you end up with a situation as what we're seeing here. So here there's, there are 2 internal states between the orange and the purple. Here there are 3. There could be 5, there could be 10, there could be 100. But what we also can show is that you only need to do the minimization over 6. Okay, so why am I saying this? Why I've got this slide? Well, the point is that when you're doing this numerical minimization, you don't have to search over some hopelessly large set. You can start your minimization right away on this topology and be sure that you're going to get the correct lower bound. And so this makes it actually practical to do. Okay, so if we go back to our system that we had, as I said before, previous estimators are going to get 0 because the observed trajectory is time symmetric. However, when we apply our sigma 2 estimator, we recover exactly what the entropy production rate is. Okay, so how might you apply this to an experimental system? So let's go to the bacterial flagella motor that we had earlier. So some strains of bacteria, they spin round and round and round in one direction, and those are clearly out of equilibrium because if you play the video in reverse, they go the other way, right? But there are some strains which they'll spin in one way for a bit, and then they'll spin the other way for a bit, and they'll spin one way. And so you take that video, you play it in reverse. It's not so obvious which way is forward, which way is reverse. And so some estimators are going to struggle with that. So what we did is we take this trajectory, we coarse grained it. We're allowed to do further coarse graining if we want into just 3 states. And we took the dynamics on these 3 states and applied the estimator. When we have a strain which doesn't switch, so this MTB24, you only get a slight improvement from going to the sigma 2 bound. This isn't surprising because the system was obviously violating detailed balance, and that was clear. But when you take a strain that switches direction, now you get a big improvement, and you can also now tell the difference between a non-switching strain at a low fuel concentration and a switching strain at a high fuel concentration. And this still works. There are details, but it still works if we coarse grain in time, if we coarse grain in space. I didn't talk about continuous versus discrete degrees of freedom. It works on continuous. Most of the things I've been talking about today are discrete, but it works in principle on both. Okay, I don't have time to talk about the other 2 examples that we looked at, but if you are interested, I'd be happy to talk about them afterwards or later or whenever. And I think maybe I have a few minutes.
Yeah, you have around 5 minutes more, and then some time for questions.
That's perfect. Okay, so great. So I want to talk about the extension to this. If you get to an even more severe state of coarse graining, so where you only have 2 states. So this is if you just have 1 state, then it's trivial. If you have 2 states, this is the most coarse grained, least but still non-trivial system that you could imagine. And this could be a gene switching on and off that you're measuring. It could be a molecule coming and docking with a sensor, undocking, just those 2 states, that's all you see. Or it could be this cow lying down or standing up, which hopefully, I will get to. But yeah, so all that you see, if you just measure 2-state dynamics, you'll see it spends some time in A, some time in B, some time in A, some time in B. And so all that you measure is the distribution of time it spends in A and the distribution of time it spends in B. And so what we're going to ask is, can we bound the entropy production rate just based on that, just based on those distributions? And so the idea is the same as before. Maybe we could do a minimization over all states, all possible systems which have the same distributions. So we take any system which has the same distribution as the one that we measure and find the minimum entropy production rate of all of those guys, and that's a lower bound. Okay. Now, I think this, again, it's going to be impractical to do this numerically. I don't know how to do this numerically. I think it will be hard. I think it's hard also to measure and to be sure that you've measured a full distribution. So we have an idea of a more practical minimization we could do. So we're going to relax this slightly, and we're only going to measure the average time it spends in A, the average time it spends in B, and also the variance of time it spends in A, or equivalently the second moment of the time it spends in A. And what we can do is we can do this transformation in such a way that we end up doing a one parameter minimization. Okay, and that's good, right? A one parameter minimization is good for the reason being that I can go away and do a one parameter minimization. So one parameter minimization is a curve, okay? And so on the y-axis here, we have the parameter that we're putting in, the variance of the waiting time. And we can read off from this curve the value of the entropy production suitably scaled, okay? And the great thing about having a curve is that I go away, I use Mathematica to figure out the derivatives and the Jacobian of the thing I'm minimizing. I do it on a cluster. I make sure very carefully that we've got the right minimization. But once I've done that, I can just save the curve, save the points on the curve. And now we can go away, we can go to our experimental system. So here I've got a gene that's just switching on and off. And this is the time it spends inactive. I can measure properties of this distribution, and I can compute this quantity, and then I can just read off from the curve what the entropy production rate or a bound on the entropy production rate is. Okay, now one of the things we applied to is this fantastic data set, Ig Nobel winning data set. So these guys put a sensor on a cow and recorded basically, is the cow standing up or is it lying down? And so you can go in and you can get this distribution of the time the cow spends lying down. You can compute this quantity and you can read off from the curve a bound on the entropy production rate, and you can confirm, as I've long suspected, that the cows are a non-equilibrium system. And this is, it's in there, it's in the waiting time distribution. And so you can read off and get this bound. Okay, so thank you. I think I'm out of time. But thanks for having me, and I'm happy to take any questions.
Cool, thanks, Dominic.
So we have questions in the chat, but also, so I'll ask these, and if there's any time left, feel free others to raise your hand too if you want to speak up. But since these came in first, I'll ask them to you, Dominic. How practical is this method as a signature of non-equilibrium behavior? And do you know how well it behaves for systems that are increasingly further away from equilibrium? Oh,
sorry, you're muted, Tom.
Did you hear me, Dominic?
Yeah, sorry, my internet cut out for a second. I didn't hear the question.
Oh, how practical is this method as a signature of non-equilibrium behavior? And do you know how well it behaves for systems that are increasingly further away from equilibrium?
Yeah, so I think increasingly far away from equilibrium is fine. I think that's good, actually. And the more a system is away from equilibrium, the more obviously the statistics will be away from equilibrium. And so, for example, if it's the waiting time distribution, you'll see very like sharply peaked distributions if the system's like way out, or very persistent cycles and so on. The difficulty when it comes to doing this is every level of coarse graining which happens, not exactly, but quite often when you coarse grain, you lose information. And so when you lose certain fields, you might not be able to see that you are out of equilibrium. And I know it's certainly the case in sometimes with these like active particle systems. I mean, I'm not the expert on this, but if you don't have access to all of the degrees of freedom, sometimes it just appears somewhat like an equilibrium system at some level. So yeah, I think the main, you need to have some good measurements. You know, the point of this is to overcome the fact that we have coarse grain measurements, but you need to have measurements of enough degrees of freedom. Does that answer the question?
I think so. I think good enough. Yeah, so we'll ask, I think so. And so we'll ask one more from the chat, and then we'll ask, and then we'll let Bill ask this question. But I'll ask this one because I think it's a good one. This is from Antonio Carlos Costa. So pretty much, so how robust is this to finite size effects in measurement data? And I guess how good is this method at measuring finite time? So if you have a finite system, how can you, how does this do?
Yeah, that's a really good question, and an important one. So this is, I said this briefly. So when I said, I don't think you can measure a distribution fully, I mean, these are a lot of information, you need a lot of measurements to measure a full distribution. If you're just trying to measure some moments, you'll get better statistics. And so for the waiting time stuff, it's, do you have enough data to measure a variance? Do you believe that you measured that properly? And also for the other data, yeah, that's why, if you have many states and you only see transitions not that often, then you won't get good statistics. So you're free to do some coarse graining of your own and get better statistics. I mean, you can also do bootstrapping, which is what we did in the paper to get bounds to be, kind of sure.
And I guess with that, can you get from the mean? If you couldn't discern the variance from that waiting time distribution, what would the answer give you if you just could discern the mean only? Would you just get 0, or do you need that variance to get?
Yeah, you need the variance. So you can devise an equilibrium system which hops between two states, and it spends a mean amount of time in both. Right, but if you want to control that more precisely, you want to, say I want to really spend a precise amount of time here, then you have to pay.
All right. Okay. So Bill, please ask your question.
Thanks. So when you talk about waiting time distributions, I guess implicitly you're assuming that successive transitions are independent of each other.
Not necessarily. So but I am assuming that it's weighted long enough, so it's fully sampled the system. We're just
sure, yeah, you're in a stationary state. I guess I don't have an intuition for whether, if I look at the transition, if I have a system that's not Markovian, or at least the variables I'm observing are not Markovian, and I measure the transitions from one state to the next, and I see or don't see evidence of irreversibility. Then if I look on longer timescales, since it's not Markovian, what I see isn't predictable from the single state transitions. So is there a sign to this effect? I mean, if I think about trying to compute the entropy of a time series, then if I don't look at long enough timescales, I'm always overestimating the entropy per unit time because I haven't kept all the correlations. But when I think about the entropy production, as I look at longer timescales, does it always come from one side or the other? Or can the longer timescales destroy the evidence for the arrow of time that I thought I had on short timescales? Or is it always the other way, that if I don't see an evidence for the arrow of time on short timescales, it could appear at long timescales?
I think I think it could be that if you don't see it on short timescales, it could appear on long timescales. I guess I was only thinking about when it is in a stationary state, and so I'm not concerned about necessarily these kind of short-term dynamics, only the long-term statistics. Yeah, I'll have to think about that.
Okay, good, thanks.
Okay, I think we are almost up to the 30-minute mark, so we should probably move on. And thank you again, Dominic. And if you have further questions, maybe you can contact the speaker separately. But Next up, we have Henry Mattingly, who's a postdoc at Yale, and he's going to tell us about chemotaxis in E. coli. So go ahead, Henry.
Thank you for the introduction, and thank you for the opportunity to speak here. My name is Henry. I was trained in my undergrad and PhD as an engineer, actually, and in the past few years have been making a foray into physics. I'm really interested in statistical inference problems and how biology solves them. And today I'm going to tell you about an exciting project I've been working on with my lab mate Keita Kamino and our collaborator Ben Makta. Okay, so biological systems routinely make decisions with incomplete information in order to survive. Depending on your perspective in this example, a cheetah is trying to catch a gazelle, and it's continuously making predictions about where the gazelle will go and then acts on those predictions. In this other example, a bacterium is trying to climb a chemical gradient of attractants and is continuously making inferences about which direction it's swimming relative to the direction of the chemical gradient, and then acting on those imperfect inferences. So my question in this project was, how does the quality of an organism's inferences affect its performance at these survival relevant tasks? Information theory is a natural language for quantifying the quality of inferences. And so I want to connect information to performance of a task. Other related questions are, what information is relevant to performing the task? What does the organism need to pay attention to? What cues in the environment? And how efficiently do organisms use the information they acquire to perform the task? So I'm going to make this a little more precise with an example. When you stream a video, say on Netflix, data is sent to your device at some rate set by your internet connection speed, measured in bits or bits per second. Your device then takes that data and renders it into a video. A faster internet speed is necessary, but not sufficient to produce a higher resolution video. Decades of work have developed algorithms that produce a video that's as high resolution as possible, given your internet speed. At some point, though, there's a theoretical limit. With a given internet speed, it's not possible to make an arbitrarily high resolution video. Algorithms that operate close to this theoretical limit use information efficiently to perform the task of making a high resolution video. And they're information limited in the sense that the only way to increase video resolution is to increase the internet speed. So we can draw a direct analogy between this example and an organism performing some survival relevant task. Rather than an internet connection, organisms acquire information from their environment using sensory organs like eyes or receptors. Then they respond to those cues and to perform the task. But the important thing is that the quality of their measurements can be measured or quantified in the same same units as an internet connection link, in bits or bits per second. So we want to know where biological systems lie on this plot and how efficiently they use information to perform tasks. E. coli chemotaxis is a well-understood task in which we can answer this question. It's actually quite convenient that Dominic was talking about bacterial motors switching directions. So to begin thinking about this, we need to have some sense of what is the goal or function of the task that the organism is performing. What is it trying to do? And then we need a quantitative measure of how well is it doing it. In the case of E. coli chemotaxis, to a first approximation, the goal is to climb a chemical gradient of an attractant as quickly as possible. So we define performance as the cell's average upgradient velocity or drift speed. To climb gradients, E. coli use transmembrane receptors and measure concentrations along their swimming trajectory. Concentration changes are encoded in the activity of key receptor-associated kinases, and then kinase activity goes on to control transitions in the cell's swimming behavior between run state in which the cell propels itself forward, and a tumble state where it randomly reorients its swimming direction. This is achieved by changing the direction in which its motors are turning. But how do we quantify information transfer in this example? This context. One of the first information quantities one might think of is the mutual information between the current signal the cell is experiencing and its kinase activity. So just note that I'm defining the signal as the rate of change of concentration, and it's log concentration because E. coli respond to relative changes in concentration over a wide range of backgrounds. The problem with this measure is it's blind to the recent past, and we know that E. coli have a short memory over which they use signals to control their behavior. What we need is an information quantity that is aware of the signal trajectory and kinase trajectory. One such measure is the mutual information rate between these two trajectories, but it also has its own problems. So to explain why, if I show you this signal trajectory, which a cell in a simulation experiences, you can tell me pretty clearly where the tumbles happened. There are these sharp changes in the signal, sharp changes in DC/dt. But in this simulation, kinase activity and motor behavior were completely independent of the signal. So the cell is not responding to what's happening in its environment. The reason there are these correlations is that the signals the cell experiences are influenced by its own behaviors. So there are these correlations even for cells that don't sense the gradient and therefore don't climb, don't navigate. So this, in which case the mutual information rate is non-zero. This is problematic. The cell needs to sense its world to climb a gradient. And if we're saying it's sensing its world but not climbing, this is weird. Fortunately, the mutual information rate can be decomposed into the sum of two directed information terms called transfer entropy rates. And it's the transfer entropy from signal to kinase activity that must be non-zero for a cell to climb a gradient, and is how we're going to quantify information transfer. Really, it measures the signal elicited fluctuations in kinase activity. Signal elicited is the important part. Okay, so this information rate, what the cell gathers from its environment, is like a budget that it can use to bias the statistics of its behavior. Ideally, it would bias its statistics in such a way that it climbs gradients quickly. This poses the optimization problem here. How does the cell maximize its drift speed given the information that it acquires? Using a mathematical model of chemotaxis, we solve this problem analytically. As you kind of could intuit, the cell must acquire information in order to have non-zero drift. This bound also depends on a prefactor f of theta, which is between 0 and 1. And this depends on parameters that characterize the cell's swimming behavior in the absence of a gradient. So there's the average run to tumble frequency, so how frequently the cell tumbles, the tumble to run rate, the persistence, how much a tumble randomizes the cell swimming direction, and rotational diffusion, how fast the cell loses direction when it's swimming. We measure these parameters from swimming cell trajectories to put numbers to f of theta. At this point, I'm going to pause and think about how much information does a cell need to climb a gradient? Naively, what would you expect? One estimate would be based on the idea that on every run, the cell needs to decide, am I going up the gradient or not? This is a binary decision that takes 1 bit to disambiguate, and runs occur every second, so 1 bit per second. But if we plot this bound, we find that chemotaxis is possible with much less information than that. In a typical shallow gradient, the cell's drift speed is a few % of its swimming speed. And this bound indicates that this is possible with hundredths of a bit per second. The reason for this discrepancy is that with 1 bit per second, the cell knows exactly whether it's going up the gradient or not on every run. And actually, to climb the gradient, it only has to know that or answer that question correctly more often than not. So it can get that question wrong and still navigate. So now we want to know, where do E. coli lie on this plot relative to this bound? And once we make a measurement, how do we interpret it? So say we find that E. coli get this much information, they climb gradients this fast. The length of this horizontal line quantifies how much information the cell acquired, but did not use to climb the gradient. These are signal elicited responses, but that did not contribute to climbing the gradient. And the vertical line quantifies the performance loss, sort of the opportunity cost of not using that information. So to quantify, to quantify how close E. coli operate to this limit, I'm going to define an efficiency of information usage, how fast the cell climbs the gradient, given the information it has, relative to the maximum possible gradient climbing speed with the same amount of information. The equation looks like this. And to measure this efficiency, we need to measure the cell's information rate and chemotaxis performance. So I'll start with the information rate. E. coli encode information about signals they experience in the activity of key A kinases. So I'm going to, so we quantified information transfer from signal to kinase activity. This can be determined from the statistics of the signals the cell experiences during chemotaxis, the kinase response function, and the statistics of noise or spontaneous fluctuations in kinase activity. My lab mate Keita Kamino designed a microfluidic device which allows him to deliver controlled chemical stimuli to immobilized cells with high time resolution. Then he can measure the kinase responses in individual cells from fret between the substrate of the kinase, key Y, and the phosphatase, key Z. So an experiment looks something like this. He delivers multiple step changes of concentration, increasing and decreasing to the same cells. This row here are the multiple responses of a single cell to repeated stimuli, and they're very noisy. Each row is a different cell. At the bottom is the population average. On the right, he computed the average single cell response to a step up in concentration or a step down in concentration. These responses have a stereotypical shape, and he fit individual responses to this phenomenological model to extract the typical cell's kinase response function. Using the same system or experimental system, can measure the statistics of noise and kinase activity. So this is done just by measuring from cells when there are no changes in concentration in their environment. These are what some of these kinase trajectories look like. And from these, you can constrain a model of the noise statistics or the noise correlation function. Okay, then lastly, we needed to measure the statistics of signals cells experience during chemotaxis. Conveniently, during chemotaxis in static one-dimensional gradients, the signals the cell experiences are completely determined by its own upgradient velocity and the steepness of the gradient in which it swims. So to measure signal statistics, we just have to measure the statistics of upgradient velocity. So we did this by measuring tracks of individual swimming cells, producing traces like these. Cells in the population have a wide variety of swimming behaviors, but we quantified the velocity correlation function of the median phenotype in this population, so shown by this red curve. Okay, so what these measurements allow us to do then is to compute this integral, which is essentially determines a prefactor between the steepness of the gradient that the cells are navigating and the amount of information they get about how fast concentration is changing as it swims. Plugging in these numbers, we found that E. coli get very little information during chemotaxis. In typical shallow gradients, they only get a few hundredths of a bit per second. These numbers are suggestively close to what I showed you earlier, but to quantify how efficiently these cells use that information, we also need to measure how fast they climb chemical gradients. Okay, to do this, we establish static linear gradients between a low concentration reservoir and a high concentration reservoir and tracked cells as they navigated these gradients. We did this for multiple gradient steepnesses and from hours of trajectory time in each condition computed the population average drift speed. As expected in shallow gradients, the drift speed increased linearly with the steepness of the gradient. All right, so now we have everything we need to compare E. coli's performance to the theoretical limit. We've measured how much information they get and how fast they navigate with that information. So what I'm going to do is plot information on the x-axis against drift speed on the y-axis, eliminating gradient steepness. All right, when we do that, it looks like this. The ratio of the blue line and the green line is the efficiency eta. And computing it, we find that E. coli climb chemical gradients at speeds of 65% of the theoretical maximum possible with the information that they acquire. Therefore, we say E. coli use information efficiently to navigate, and their chemotaxis is information limited. All right, just to summarize, we asked how well do organisms use the information they get from the environment to perform tasks that are relevant to their survival. Answering this in E. coli required measuring the rate at which a biochemical network transfers information for the first time. And we found that E. coli get very little information, but they use it efficiently to climb chemical gradients. These results suggest that perhaps organisms are under selective pressure to use the information they get efficiently to carry out survival relevant tasks. I want to thank our funding sources and my collaborators, and thank you for listening. Take any questions.
Thank you, Henry. So if you have any questions, feel free to raise your hand. I actually had a question. Wait. Okay. I actually had a question for you. So with bacteria, some of them like hedge their bets, so they don't, they do different things, and some of them will survive, and those that do survive, they win big. So I was wondering, so with this low information rate, do you think that it's actually them being efficient, or that they're just taking a little bit of information and they're hoping, they're hedging their bets that they get the right amount of the right information out?
Okay, so there are definitely differences in a population in terms of how much information an individual cell gets and how efficiently it uses that information to navigate. I think that one way of thinking about hedging bets is that climbing gradients is not the only thing that E. coli care about or use information to do. Oftentimes people pose or have in the past posed the trade-off between staying near a local maximum of concentration versus navigating towards that maximum. And yeah, I think it's totally reasonable to think about why an why E. coli are not closer to this bound say. There may be other things they're trying to do that would explain that.
Okay, thank you. And is there any other questions for Henry? I don't see anything I don't see anyone raising their hands. And you can write it in the chat if you feel more comfortable.
Okay, if there are no other questions, I think we can thank Henry again and move on to our next speaker, who's Rosa Martinez-Corral.
Thank you very much, and thank you very much for the invitation. I'm very happy to be here My name is Rosa. I'm a postdoc in the Department of Systems Biology at Harvard Medical School, working with Professor Jeremy Gunawardena and Angela DePace. I was originally trained as a biologist, and then I became interested in using tools from mathematics and physics to understand how cellular behavior is regulated and try to understand general principles about cellular regulation. So today I want to talk about transcriptional control. So as you probably know, not all genes in the genome of a cell are expressed at all times, but depending on the environment and the cellular history, some genes are kept off and some genes are transcribed to certain levels. This process is orchestrated by transcription factors, which are proteins that are typically produced. So yeah, I was talking about transcriptional control and how this is an important process for cellular regulation. And for this process to occur, transcription factors are key. Transcription factors are proteins that have the ability to bind on the DNA in a sequence-specific manner and directly or indirectly recruit or regulate the activity of the RNA polymerase complex, which then is in charge of transcribing the DNA into RNA. Because these transcription factors are typically produced or activated in response to particular signals, then they provide this link between the information in the environment and a given gene expression pattern. In eukaryotic genomes, which is the focus of my research, a given gene is typically controlled by many transcription factors of the same and different type that together regulate the gene. And so this raises the question of how the combinations of transcription factors together regulate a given gene, or in other words, how is the information contained in the type and levels of transcription factors integrated into a given gene expression pattern. And for simplicity, for today's talk, we are only going to focus on what's the relationship between transcription factor concentration and gene expression response. A lot of work has been focusing on the role of direct protein-protein interactions between transcription factors to explain how they together regulate transcription. However, the problem with this view is that in principle, one would expect that if there are rearrangements between binding sites or some sites disappear, then the expression pattern should be lost. However, there is a lot of research that shows that this is not the case in eukaryotic genomes. We can have substantial binding site rearrangement and still maintain a given expression pattern. So what I want to describe today is some work we've done that illuminates on some alternative mechanisms by which transcription factors combinatorially regulate transcription. For transcription to occur, polymerase has to undergo through a series of transitions. For example, it first has to bind on the gene, then it has to be phosphorylated in some cases, and then it can proceed to elongate and produce the mRNA molecule. And these different processes can be regulated by transcription factors in different ways. So let's imagine that we have transcription factor A that is enhancing the first step, and transcription factor B that is enhancing the second step. Then you can easily see that if only one of the two TFs is present, then expression will be much less than if the two TFs are present together, right? But the point of this type of synergy, which we call kinetic synergy, is that it emerges as a result of the functional interactions between the TFs, not because they are recruiting each other on the DNA. And so as a result, we can obtain this behavior even if the two TFs bind on the same site. Because TFs are constantly binding and unbinding, then as a result of their functional complementarity, it is enough for this kind of configuration to be able to produce synergy. And I don't have time to go into details today. If you want to read more, here is the reference. But I wanted to highlight this work because I think it's important to keep in mind that not everything has to do with binding interactions between the TFs. There are also these functional interactions that are very important. But for the rest of the talk, I'm going to focus on binding interactions. And we will simplify this view of the transcription cycle and focus only on the relationship between the binding configuration and expression. So we will assume that depending on what is the configuration of the regulatory sequence of a gene, we have a given gene expression pattern. And so by focusing then on what is a given, what determines a given configuration of binding, if we can then describe what determines a given distribution of binding configurations, then we can link this to a given gene expression pattern. Using this type of approach, a previous study in the lab focused on the study of a Drosophila gene called Hunchback, which responds to a transcription factor called bicoid. If we plot this input-output function, as you see here, bicoid activates Hunchback in a highly nonlinear manner, where small changes in the concentration of bicoid lead to large changes in the response. This is believed to occur from bicoid binding to 6 sites on the regulatory sequence of the Hunchback gene. And so what this previous study asked, or one of the questions that it asked is, what types of interactions do we need to assume between bicoid molecules to explain the generation of this response? And one of the conclusions was that it was not enough to consider interactions between one molecule binding at one site and another molecule binding at another site, but that it was also important to consider these higher order interactions where the binding of a molecule at one site is influenced by the binding configuration of more than one molecule at other sites. And so this motivated, in general, the question of how can these higher order interactions emerge, and whether there is any mechanism that can generate flexible higher order interactions between binding sites that are not in direct contact with each other. And this is what I want to explain now. But first, I want to tell you about the language that we use to define these higher order interactions in a biophysical manner. And then I'll tell you about how allosteric systems can be a mechanism to generate these higher order interactions. So in the Gunawardena group, we used the linear framework approach to study gene regulation. The linear framework is a graph-based approach to work with Markov processes that uses graphs to describe a particular system. So for example, in this case, where we have an example of a linear framework graph that describes the binding of a transcription factor to 2 sites on a regulatory sequence. The nodes of the graph are the binding states, the edges of the graph are the transitions between states, and the edge labels are the rates of these transitions. If we assume that there is a certain probability of the system being in each configuration, then the probability of finding the system in each state evolves according to this master equation, which is given by the edge labels of the graph and is linear in the vertices, and therefore that's how the name linear framework arises. For today, I'm going to focus on steady state behavior. For systems, when considering the steady state of systems, we can distinguish 2 types of graphs. If the cycle condition holds, meaning if we take a cycle like this one and take the product of the edge labels in the clockwise direction, and this equals the product of the edge labels in the counterclockwise direction, then when the system reaches steady state, it will satisfy little balance. And therefore, when the cycle condition holds, the linear framework formalism is representing equilibrium statistical mechanics systems. In contrast, if the cycle condition doesn't hold, then it will represent a non-equilibrium system. So with the same formalism, we can study both equilibrium and non-equilibrium systems. For today, I will only focus on systems that satisfy the cycle condition and therefore that relax to an equilibrium steady state. To calculate this steady state of the system, we use an approach which works as follows. So we define a node as a reference, so for example, this one, and then we need to calculate these mu quantities, which are just ratios of forward and backward transitions over a path from the reference node to the node of interest. So for example, for mu4, we would get this over this times this over this. We do this for all states, and then this gives us the, if we divide the mu for a given node by the sum of all of them, this gives us the steady state probability of each node. For equilibrium systems, this is completely equivalent to the thermodynamic formalism. So many of you may know these thermodynamic models of gene regulations that are based on these statistical mechanics principles that so they enumerate all the possible configurations of the system, and by assuming that the system reaches equilibrium, then the steady state probability is given following the Boltzmann distribution. So our formalism is equivalent to this, and you can see this by noticing that at equilibrium, the ratio between the forward and the backward transitions between two states is related to the free energy difference between these two states. However, we think there is an important advantage to working with these graphs, which is that the focus is on the transitions rather than on the states. And if we are interested in understanding binding and unbinding processes, we think that this is an advantage. Because at equilibrium, only the ratio between the forward and the backward transitions matter, from now on, I'm going to represent the graphs as equilibrium graphs, where we only represent one of the transitions, even if it is reversible. And the label now is going to be the ratio between the forward and the backward transition for the corresponding individual reactions. So our example graph reduces to this graph. Again, it's reversible, it's just that I am only showing one edge. And now there are two types of parameters here that are important. We have on the one hand the binding affinities, and on the other hand the binding cooperativities. The binding affinities are just the ratios between the transcription factor binding on rate and off rate. And we use this notation. So we have binding to site I when the sites in S are occupied. So for example, here we have binding to site 2 when site 1 is occupied. If there is nothing bound, we talk about the bare affinities, and this would correspond to these two parameters here. If we divide the binding affinity for a given site when something is bound to the binding affinity to that same site when nothing else is bound, then we obtain the binding cooperativities. These are the parameters that tell us by how much the affinity changes when these sites in S are occupied. So as a result, we can completely parameterize a binding graph with just the bare affinities and the cooperativities, because we can rearrange this as you can see here. If the cooperativity is less than 1, what this means is that the sites in S produce binding. If it is equal to 1, it means that the sites in S don't do anything to the binding to site I. And if it is greater than 1, it means that the sites in S help bind. The number of sites in S is what we call the order of the cooperativity. So if there is only one site, we talk about pairwise cooperativity, but we can also describe these cooperativities for higher orders of interactions. So for example, here we have the cooperativity between two sites and a third one. So with this, you see how we have, so this is the language basically that we use to talk about these higher order interactions in a way that is biophysically meaningful and that also tells us very clearly what is happening between the interactions between sites. And so now the question was, what can be a mechanism to generate these higher order cooperativities? And for this, we turn to the phenomenon of allostery. So allostery is a widespread phenomenon in biology where the binding of a ligand to a macromolecule at one site influences the binding of a ligand at another site of the molecule that is far away. And this typically happens because of some conformational redistribution. One of the molecules where this phenomenon has been studied a lot is hemoglobin, which has 4 sites for oxygen binding, and the binding of oxygen at one site helps oxygen bind at other sites. In order to explain this phenomenon, Monod, Wyman, and Chen proposed what is known as the Endao model, which assumes that the molecule is changing between two conformations, one that is low affinity for the ligand and the other one which is high affinity for the ligand. And so the system is in equilibrium between these two conformations, but when oxygen binds or the ligand binds, it kind of stabilizes the high affinity conformation so that more ligand molecules can bind. The same model was used by Leonard Milne to suggest how transcription factors could bind cooperatively on the DNA, even if they don't directly interact with each other. Here, the two conformations would be given by the site, the regulatory region being occupied by a nucleosome or not. So in case you don't know what a nucleosome is, it's just a core of histone proteins that are around which the DNA is wrapped in eukaryotic genomes. And so these cores of histones can sometimes unbind. And when that happens, then the TFs could have a higher affinity for binding, and then this would reduce the nucleosome probability of binding again, and so it would help more transcription factors to bind. And so with this type of model, we can obtain nonlinear input-output relationship between transcription factor concentrations and the occupancy of the DNA by transcription factors. So we wanted to know what are the higher order cooperativities of this system and whether their patterns are flexible or they are constrained. For this, what we do is we represent the NWC model in terms of an equilibrium graph. Remember, everything is reversible, but I'm only showing edges in one direction. For simplicity, I'm going to remove the transitions between states that have something bound, and I will show the graph like this. But there are transitions between all the possible states between the two conformations. This graph is parameterized in terms of three parameters: the affinity for binding at one conformation, the affinity for binding at the other conformation, and the transition between the two. What we have found is that we can coarse grain this graph into a new graph where the conformations have disappeared. And now what this graph is telling us, it's telling us just how the TF is binding at a particular site, but it's going to be some sort of average binding across this conformation. So we call this an effective binding graph. Just to be clear, what we are doing here is we are coarse graining together the states that have the same binding pattern into a new state. We have developed a method to assign labels to this coarse grain graph, such that the steady state probability of the nodes in the coarse grain graph corresponds to the sum of the steady state probability of the nodes in the detailed graph that get coarse grained together. The way in which we assign these labels is the following. So, for example, if we are taking this label, we need to calculate the μ's for the nodes that get coarse grained into the target node and divide that by the μ's for the nodes that get coarse grained into the source node. And because all the binding affinities are the same at a given conformation, we will obtain the same for all the different sites. And in this case, the expression, the formula is going to be the same. We can do the same procedure to calculate the effective binding affinities when something is already bound. And then if we divide these effective affinities by the bare effective affinities, we obtain the effective cooperativities, which is what we want. We wanted the effective higher order cooperativities of the NWC model. Now I'm going to do some rewriting of this formula. For this, let's focus on the subgraph that is composed only of the two states that don't have anything bound in between the two conformations. If we would consider this as a graph on its own, remember it is reversible. I'm just showing the edge in one direction. Then the probability of each of the two nodes is what you see here. And now let's use this notation to represent the average of a function over this probability distribution. It turns out that the formula that we calculated before for the effective binding to a site when nothing else is bound is the average of the affinity per conformation over this probability distribution. And the same for the other effective affinities, they can be represented in terms of these averages of powers of the affinities per conformation. And what this allows us to do is that we can represent we can calculate these effective bindings for any number of conformations and for any number of sites. And this lets us arrive at this formula for the effective cooperativities, where S is the number of sites that are occupied. And what this formula is telling us is that for this NWC model, the higher order cooperativities are highly constrained because they always increase with increasing order. So what we wanted to know is whether there are options, there are other ways to arrange this, the affinity patterns in the detailed graph to obtain arbitrary patterns of higher order cooperativities. And to do this, what we found is that we need many conformations, and we need that each site in each conformation has a different affinity. In contrast to the fact that in this case, all the sites at a given conformation have the same affinity. I forgot to say, but in this case, it doesn't matter how many conformations. This constraint will always be there. However, if we allow the sites to be different and we have enough conformations, then we can have arbitrary effective cooperativities. To exemplify this, let's consider an effective graph that represents the binding of a ligand to 4 sites. It is parameterized in terms of these effective affinities and effective cooperativities. And here I'm showing just one parameter set that completely parameterizes the graph with the corresponding input-output relationship between transcription factor concentration and average fraction of sites occupied. And so the question is, can we derive a detailed graph such that when we coarse grain it, it gives us these parameters? And I won't go into the details, but what we show in the paper is that, yes, we can do that. In this case, we need 16 conformations, and we need that each site, that each conformation has a different affinity between them. And then we present a way in which we can coarse grain this, in which we can calculate the parameters for the detailed graph that will give us the coarse grain parameters. As an example, here we're showing 3 sets of parameter sets and the corresponding parameters for the detailed graph that would generate those effective parameters. And here on the right, you can see the corresponding input-output relationships that we obtain. And you see that different patterns of effective affinities and effective cooperativities can generate a wide variety of input-output relationships. So this flexibility result is telling us that allostery could be a powerful way in which to generate complex input-output relationships between transcription factor concentrations and gene expression responses. All of this has been based on just one type of transcription factor, but we could also extend the formalism to more than one TF. And so we think that this, in combination with the functional interactions that I also mentioned at the beginning, plus the effect of non-equilibrium processes such as phosphorylations, all of this has to be taken together when thinking about how transcription factors combinatorially regulate transcription. And all of these is things that we are very interested in exploring further. To finish, I just would like to thank my advisors and collaborators, funding sources, and you for your attention. I'm sorry for the mess at the beginning.
Thank you, Rosa. Is there any questions for her? Marianne, please unmute yourself.
Okay, great. Thanks so much. I really enjoyed this talk. This was fantastic. I just had a quick question. So I really liked your graphs of fractional saturation versus concentration. And I was wondering, if you see such a graph, can you infer back the rates, or is that sort of too hard a problem?
No, that's a very good point. I didn't go into this, but we mentioned this in the paper that we can obtain the same curve for very different sets of parameters.
Interesting. Okay.
So, I mean, we haven't explored it in detail as to whether there are particular curves that are only uniquely determined by, at least for a sigmoidal, you can have different sets of parameters that will give you the same curve. So definitely it's telling you you need to know something else.
Okay, thanks so much. Great talk.
Thanks.
Is there any other questions? Raise your hand. There's one in. Oh, Gustavo.
Hello, can you hear me?
Yeah.
So it's a question related to that one. Do you have a way of calculating, given a number of sites and a known input-output relationship, do you have a way of calculating how degenerate the solution might be? So how many combinations do you have?
No, we haven't addressed it in this way now. No. But I can tell you that it's many.
So what would be interesting is to be able to quantify that, because then it also gives you some information about how evolution can lead to a certain solution. if it's an easy solution, if there's many degenerate possibilities, or if there's just some very unique possibilities.
Yeah, I can definitely, yeah, definitely. Yeah.
Oh, from that,
But if-
oh, I'm sorry if you. I was just gonna actually follow up on that. I thought that with your graphs, you could just enumerate, you know, how many of the same graphs you have, and that would just go into like a constant in front of your solution. Is that not what you're talking about?
So the thing is, if we have a curve like this, the questions were, do we know this? And the answer is not, because different patterns of parameters can give you the same curve. Our point was more about these parameters are telling us something about the interactions between the sites and about the conformations that you need. And so if you would know this, then you could know kind of what's the minimum number of conformations that you need, and what type of interactions of affinity patterns you need to generate them. But going from here to here, no. I mean, we've done work on the sharpness of sigmoids and how to get very sharp curves. So like curves that switch quickly between a low value and a large value. And what we find is that for systems that are at equilibrium, the Hill function is like the limit of those curves, but still you get many parameter sets that can get you the equivalent of a Hill function.
Okay, thank you. I think that answered, or at least my question from it. And I think we're going close to our break, so I think I'll give it to Kamesh to close us out. Thank you.
Thanks, Trevor. So yeah, if there are no more questions, let's thank all the speakers again, and we'll be back at 1 o'clock for the next session. And there's a half-an-hour break. You can go stretch your legs, get some refreshments.
So welcome to the second session. We are going to follow the same rules here as we did last time. So please post your questions in the chat. If there's something very pressing, we will read it out during the talk, but otherwise we will do questions at the end because interruptions are a little bit tough in a Zoom session like this. All right, and so getting to our first talk, we're going to have Yuhan Wang talk from UIUC talk first. She's going to tell us about protein-protein interactions in living cells.
Hi, everyone. My name is Yuhan. I'm from Grubler Group, University of Illinois, Urbana, Champaign. Today, I'm going to talk about quantifying protein-protein weak interactions in living cells. All right, so our group are interested in studying protein dynamics in living cells. Studies have been done from different perspectives. We've studied protein, how the cell volume perturbation changes protein folding in cells. We've studied how the cellular environment affects protein RNA binding inside the cell. Now we're exploring how the cellular environment play a role on protein-protein interactions in living cells. Before we start, let's think about what we have in a cell. In a human cell, we have ions, proteins, nucleic acid, etc. These molecules form a complex network of the interaction, complex network of interactions inside the cell. Not only for those strong interactions, but also for those weak interactions. Weak interaction means interaction with a free energy around several kBt. They're harder to detect comparing to strong interactions, but they are just as important in the cellular process. So we have three concepts of weak interactions: crowding, sticking, and quinary interactions. Today, we will mainly focusing on the first two. Crowding means volume exclusion. It is due to the short range repulsion among micromolecules. Other than volume exclusion, proteins and RNAs, they're both charged. They can interact by electrostatic, hydrogen bonding, hydrophobic interactions. So we can group all these interactions together and call them sticking. With crowding and sticking, cellular environment will not as simple as a test tube. So we want to carefully pick our model system to make our study easier and better. So the model system we pick is called beta-AB in living human cells. Beta-A and beta-B are two tubulin-like genes from bacteria. Similar to human tubulin proteins, A can bind with B, form heterodimer, and then those heterodimer can further form protofilament or fiber-like structures like this. The good thing of using beta-AB is that they're originally from bacteria, so they're orthogonal to human native tubules. When we study them in the human cells, we can consider the cell as a substrate to quantify their association and dissociation, which means beta-AB can respond to the environment, but at the same time, they're having some independence to the cellular environment. So to detect them in the cell, we can label them with fluorophores and then using FRET. After FRET labeling, we can get those TM images, which show that with the FRET labeling, they're still able to bind and form tubule-like structures in vitro, those tubules. Now the question is, they can bind, but how can we quantify their binding affinity? In vitro, in cell, and by simulation. Let's look at them one by one. The first thing, in vitro. In vitro, we usually quantify binding using titration. We have beta-A labeled with green, B labeled with red. So when there is only green in the solution, we will get a spectrum that has a green peak, obviously. And then this can be the original point here. And as we increase the concentration of the red protein in the solution, more and more red will bind with the green and having FRET. Because of FRET, we will get an increasing of the red peak and a decreasing of the green peak until all the green are used up in the solution. So if we plot the increase of the red or the decrease of the green, here we can get, and as a function of the protein concentration, we will see the binding curve increase fast in the beginning and then go saturate. From this curve, we can fit the dissociation constant KD, and this KD will represent the binding affinity of the beta-AB in vitro. This is how we do it in vitro. Now moving from in vitro to in cell, can we use the same method as in vitro? Can we plot the same curve as this one? The answer is no, because in vitro, we can always control the green protein concentration as a constant. But the concentration, when it comes to in cell, it varies from cell to cell. So instead of having one single curve like in vitro, we will end up having multiple curves and we will not be able to fit. So to solve this problem, we add one more dimension to the curve. So even there are multiple curves to fit, all the curves will end up on a same surface in the 3-dimension. So by fitting the surface, we will be able to get the in-cell KD. So we move from a 2D curve to a 3D surface. Now let's take a look how we do this in cell. Actually, we can't precisely modulate the in-cell concentration, but we can precisely control the ratio. That's why we choose microinjection instead of protein expression inside the cell. What we do is we pre-mix the protein A and B with a certain ratio, and then we micro injected the mixture into the cell. Under microscope, we will get a fresh signal of the cell, and then we can plot this intensity as this one single data point. And we do this for multiple cells because all the those ratios are determined. So all those points will be end up on the same plan. And then we change this ratio to a different one, and we will get another plan. In the end, we will get all those data points. And we can fit those data points into a 3D surface like this. So by fitting, the KD, dissociation constant, can also be obtained from the fitting, which is 2.3 micromolar for the in-cell binding. We can even take a look at the 3D surface and then make a projection. So project it to the XZ plane or the YZ plane, and we can just get several 2D curves like in vitro. That's how we do it in cell. And to be more comparable, we can use similar ideas to determine the in vitro KD again with this 3D fitting. So we will have more data points because we are changing the concentration of BA as well. So the in vitro KD, we can get is 17.2 plus minus 11 micromolar. This is larger than the in-cell KD. So the smaller KD means a stronger binding. This showed the in-cell KD, the in-cell binding is stronger than the in vitro binding. Cellular environment is promoting the beta-AB binding. Now we want to ask, which part of the cellular environment is making contribution? So we can change the in vitro condition to mimic cellular environment. So currently in vitro, we are here, and we want to mimic a condition to here. So in vitro, we can add lysis buffer to the system to mimic the high salt concentration condition, and we can add phycon molecule to the system to mimic the crowded environment, and we can add diluted human cell lysate to mimic sticking environment, and the result will be like this. So it shows that the salt is not the main promotion, and both crowding and sticking is bringing the KD down to a value very close to the in-cell. It seems crowding and sticking are both contributing. However, if we take temperature dependence into consideration, at a higher temperature, which is the body temperature, 37 degrees, the in-cell KD will increase from 2.3 to 4.8. All the in vitro binding also has been weakened. However, the crowding is not presenting a significant temperature dependence. It only changed slightly, while sticking is the one that showed the best trend as the in-cell. Now we're thinking of whether we can simulate this binding process. So in simulation, we can simplify the system by considering protein A and B to two spheres, and they are doing Brownian motion in the space, moving around. The potential energy between them will have two terms, a repulsive term for a short distance and an attraction term for a binding distance. And those proteins are labeled by Fret. So when they're not binding, they only emit green fluorescent. When they bind to each other, red fluorescent will be triggered. So under this potential field, let's run a Brownian motion and see how things goes. We can see it start moving around, and then let's play it faster. And we can see there are complex start forming from here, 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 and maybe here. So this system is starting from a random position. And it end up forming some complex structure or dimer structure in the end. The system will get equilibrium after a while, and both the green and the red fluorescent are stable. This is from random position, and we can even run a reverse motion by starting from all the proteins are initially forming dimer, and then they are diffused around to dissociate. And we will end up getting the same equilibrium state. For the simulation, the same idea, we can vary the molecule concentration in the system and vary from 0 to 24 degrees, just as in vitro and in cell. And we can end up getting a 3D surface like this. Or we can even add crowders to the system to occupy some space to simulate the crowding environment, or adding additional attraction between this third molecule, we cannot call it crowder again, the third molecule to protein A and B to simulate the sticking condition. So we can get those two plots, and we can see the curvature difference between the two plots. That is the binding difference between the two conditions. And this part of the project is still ongoing for getting more quantitative data. To quickly wrap up, with FRET labeling, the beta-AB form tubules in vitro. In vitro KD can be quantified using titration, while in cell, KD can be fitted from the 3D binding surface. Comparing to in vitro, cellular environment is promoting the beta-AB binding, and this promotion is contribute both from crowding and sticking. That's because crowding is reduce the dissociation rate, and sticking is increasing the association rate. So both of them end up promotes the beta-AB binding in cells. Before we stop here, there is one more thing I would like to share with you guys. Some of you might have noticed in the simulation video, in the end, A and B molecules not only form dimers, but also form trimers or even more. For example, like those group of molecules, they form trimers or sometimes even 4 molecules together. So currently in this simulation, we haven't specified the binding site between AB surface, so they are bind non-specifically. But once we give the protein an orientation to bind, a large chance is that they will form tubule-like structures, just like those in vitro TEM images. And the good news is that not only in vitro, we find evidence of beta-AB form microtubule inside the human cell. So this will lead our studies of quantifying beta-AB binding from dimerization to microtubule formation in the future. So I will stop here, and I would like to give big thanks to my advisor, Martin Grubler, and then my lab mates who helped me in this project, Mahima and Brooke. I would also like to thank our funding agency and all other Grubler members for being supportive. Thank you all for listening. I'm happy to take any questions.
All right, thank you, Yuhan. Hi, everyone. My name is Endao. Yeah, I'm a postdoc at CPBF, and yeah, I'll be monitoring the time and questions. So I haven't seen any question in the chat yet. So oh, okay. Now I have a question from.
Is the binding unbinding out of equilibrium? Could you specify what do you mean by out of equilibrium?
Yeah, please feel free. Oh yeah, go ahead.
So because you said that the KD is different in vivo or in vitro or in cell, so it is a little weird to me if so for example, energy source like ATP is not involved in this kind of reactions. So in the simulation, is your simulation like a completely passive simulation, or is there an energy source that can change this binding unbinding kinetics?
So in cells, the binding and unbinding out of the equilibrium is shifted due to the equilibrium in the cell is shifted due to the in-cell crowding and interaction with the third protein, not with itself.
So it's still equilibrium.
Yes, so it still reaches equilibrium in the end.
Okay, thanks.
I find another question. When you calculate KD from FRET, does the formation of the tubular structure matters? Yes. Here, when we calculate KD from FRET, we are assuming they are forming dimers. But if we will include trimer or even polymer into calculation, then we will need to calibrate our formula to a more complex form. But currently we are doing a dimerization calculation.
Okay. Bill raised his hand.
Yeah, I was just curious, when I understand that you do different things to generate crowding and stickiness, but at some level it's hard to have lots of sticky things around if it's not crowded, right?
Yes.
So is the sticky condition at actually lower concentration, lower volume fraction than the crowding condition, so that you can actually separate these?
Yes. So the crowding is the crowding we do in vitro is 150 mg per ml, and the cell lysate we're adding to the system is only 2 mg per ml, which is much lower than the crowding condition.
Okay.
Yeah. So it's a diluted cell lysate.
And should I think about the effects of either crowding or stickiness that right, when I think about binding, the chemical potential is actually determined by the concentration of molecules free in solution, right? So if there's places for them to stick, then the concentration that you measure is the total concentration, and then some fraction of them are stuck. And so should I think of it that the reason the binding constant changes, but then, sorry, then I don't understand why the binding constant, why the binding becomes tighter.
Oh, for the for the sticking condition, that is because in the sticking, the crowding, the A can bind with B, but A is also bind with the third particle, and B can also bind with third particle. So they can find each other on the surface of the third particle, basically. That will help them to easier find each other and bind. That's why the sticking is promoting the binding. So we can see this on the simulation, like when there is a third particle in there, they will grouped around the third particle and form a trimer or like dimer, whatever, and have FRET.
Whereas crowding, I should just think of it that it takes away volume, right?
Yes. So we are also doing a collaboration with Murphy's group of doing a nanoparticle. So we will have a third particle, which is nanoparticle in the solution that is much larger than the protein. And the two proteins can bind on the nanoparticle surface. They can find each other on the surface of the nanoparticle. Which is,
thanks.
Another collaboration.
Oh, and by the way, well, your question is highly non-trivial, if I chime in for a second. We've done measurements on RNA protein binding, same kind of experiment, and get the exact opposite effect, what you were thinking. So there can be situations where the competitive binding actually reduces the KD in cell. But in her experiment, the exact opposite happened with the tubulins.
So to follow up on that question, so in your simulation, when you have the white particles, what's the purpose of those particles? Are they just there to occupy space, but without interacting with the green and red particles?
it depends on what kind of situation you want to simulate. If you want to simulate crowding, then they're just occupied space. They don't have any interactions to the green or the red. They're just crowders that occupied space. If you want to simulate sticking, then we will, of course, reduce the number of them in the solution. They will be much more dilute, but they will have some interaction with the green molecule and the red molecule because they're sticky.
Okay, and the size and the size of the particles are based on experiments, or you just use like same particles of same size.
Here I use the same size because even in the experiment I choose a phycol. The phycol has different size. I picked the size that is similar to the protein size. So I use phycol 70, which is very similar to the protein size. So in the simulation I'm also using the same radius.
I see. Very nice. Thank you. Oh, we have one more question now. From Yaojun. Yaojun, feel free to unmute yourself. And,
if we think of the KD value as a reflection of the intercellular environment, would you see different KD for different cells, or say in cytoplasm versus nucleus? So here, yes, we've seen difference in different experiments. For RNA protein, we've like different for cytoplasm and for nucleus. And for my experiment, beta AB, because they are proteins located in cytoplasm, so I don't have a comparison of the nuclear to the cytoplasm. And the cell-to-cell variation is also exists in there, but it's not a large variety. I mean, it's within the error. You can see the error is 2.3 plus minus 0.7. That include the cell-to-cell variation. Sure. But we haven't tried for the tubulin yet. That's only for dimerization, quantifying dimerization. We are further interested in trying how to form polymers, how to form tubulin-like structures.
all right. So yeah, I don't see more questions. Let's thank Yuhan.
Thank you, guys.
Yeah, thanks again. That was a really interesting talk. But let's move on. Our next talk is from Sihan Chen from Rice, and he's going to be talking about something about active gels.
Hello, everyone. My name is Sihan Chen. I'm a graduate student at Rice University, and I work with Fred McIntosh. And I'm doing theoretical studies by polymer networks. And today I'm going to talk about motor-free contractility in active gels. This is a theoretical mechanism that can generate contractility without motors. So contractility is everywhere in living systems. For example, we can move because of the contraction in our muscles. This contraction is caused by myosin motors, which pulls the actin filaments inside muscles. And to show how strong this contraction can be, here is a video of a reconstituted actin network and myosin motors. We can see an entire network is contracted to these small clusters. And there are many other proteins which works in a similar way as myosin, and they are called molecular motors, because although they are very small, they can do exactly everything a real motors can do. And these motors are responsible for most contractility and also force generation in living systems. However, recently there has been some experimental evidences for contractility without motors. And I'm going to show you one of them. This structure is called actin-myosin ring. It is a ring-like structure formed by parallel distributed actin filaments and myosin motors. It is formed in the middle part of the parent cell during cell division, and the contraction of this ring divide this parent cell into two progeny cells. For a long time, it has been believed that myosin is responsible for this contraction. However, some recent experiments find out that this might not be the case. For example, this is an experiment on Drosophila embryo, where people have measured how the perimeter of this contractile ring changes with time. And if you look at these two black curves, they are the contraction of regular cells. Then what they did is that they tried to block motor activity by knocking out some motor-related genes. So they did that for two different genes, and the results are the green curve and the red curve. So these two curves are contraction of of cells without motor activity. And surprisingly, what they found that is that although the starting of the contraction is affected, after 40 minutes, the contractile velocity with or without motor activities are almost the same. It means that motors may not be needed after 40 minutes. And there are many other experiments like this on both animal cells and even bacteria that show evidence for motor independent contraction. And these experiments inspired us to propose a motor-free contractile mechanism. Before I introduce our mechanism, let me first talk about how motors work. This is a small video of how myosin works. You can see myosin can walk on actin filaments. The interesting thing is that they only walk in one direction. And one reason they can do this is that there's an energy source coming from ATP hydrolysis. This is important because the active binding and unbinding can break detail balance. And without it, everything is in equilibrium and nothing happens. So activity is the first ingredient of directional motion. However, activity itself is not sufficient because these motors are very small. And on this length scale, everything fluctuates. And then activity just becomes an unidirectional fluctuation. To turn this fluctuation into a directional motion, a breaking spatial symmetry is also needed. To show how this works, let me introduce a toy model proposed by Richard Feynman. It is called Brownian ratchet. A ratchet is something formed by an asymmetric gear, this is part one, and a paw, this is part two. And you have probably seen or used one of the real ratchets in your daily lives. It is a tool that only rotates in one direction. This Brownian ratchet is very small, so thermal fluctuation is important. And with a finite temperature, it starts to do random fluctuation. However, if these part one and part two have the same temperature, everything is in equilibrium, and we don't expect any motion, any average motion. However, if T1 and T2 are different, then there starts to have activity. And this asymmetric gear can rotate both in the counterclockwise direction or the clockwise direction. However, because of its asymmetric shape, it is more likely to rotate counterclockwise. So on average, there's a directional motion in the counterclockwise direction. And so this toy model shows how activity plus spatial asymmetry gives directional motion. And this is exactly how motors work. For motors, this spatial asymmetry comes from the polarity of the substrate filaments. Most biopolymers, including actin, are polar, and they have well-defined plus ends and minus ends. And this asymmetry can be modeled by an asymmetric binding potential. You can see that the shape of this binding potential is very similar to a Brownian redshift. And when the motors actively bind and unbind in a way that breaks detail balance, it leads to a directional motion of the polymer. So polarity is the asymmetry for motors, but it is not the only asymmetry for biopolymers. There exists another asymmetry. That is the asymmetric force extension relation, or so-called the nonlinear elasticity. To show this, let me use an example with my camera. So here's a rubber band in my hand, and I can either stretch it or compress it. But when I stretch it, I feel a very strong restoring force. And when I compress it, I feel nothing. Because when I compress it, it buckles. So there's an asymmetry between stretch and compression. And this is exactly what happens for biopolymers. Most biopolymers are semi-flexible. They have stronger bending rigidity than most synthetic polymers. And this asymmetry, hard to stretch and soft to compress, also exists in biopolymers. And this asymmetry becomes an asymmetric function if we plot the force extension relation of a polymer. This asymmetry is generic for most cytoskeletal polymers, and it even exists in DNA filaments as well. And with a finite temperature, this asymmetric elastic energy leads to an asymmetric Boltzmann distribution of the polymer rest length L0, together with a thermal fluctuation with a length scale delta L. Using this asymmetry, we are able to build a motor-free contractile mechanism. And as we will show later, our mechanism is robust for any asymmetric relations. For example, both the semi-flexible relation or the simple linear two-spring relation both generate contraction. So to realize this mechanism, we build up a minimal model in which we consider a semi-flexible polymer networks connected by transient cross-linkers that can bind and unbind on the network. Here we focus on a particular polymer segment, this purple one in the network, and we treat the rest of the network as a substrate, this gray one here. And this substrate, we assume it to be viscous just for simplicity. We also have results for viscoelastic substrates. And we also assume this substrate to be apolar. Here, this apolar mean is not required by our model. We just use this as a worst case scenario because no motor activity is possible on apolar substrates. And this apolar means that either the network is isotropic or it is formed by apolar filaments such as intermediate filaments. And this purple polymer can actively bind and unbind on the substrate if some energy source is involved. And we assume that the substrate has a rugged binding potential. So when the polymer binds to it, the binding potential can change the polymer length to a binding length LB. Because we assume the binding potential, the substrate is apolar, the binding potential should be symmetric. So this distribution of LB should be asymmetric around L0, the polymer rest length. Here we choose it to be a simple uniform distribution over a width D. This D is a length scale of the binding potential and can be regarded as the spacing between binding sites. we should point out that our contractility is robust for any symmetric PB, and even for a slightly asymmetric PB. And this distribution PB suggests that when the polymer binds to the substrate, it is either stretched or compressed with the same probability. However, the first extension relation is asymmetric. This means that when the polymer is stretched, it provides a strong restoring force, but when it's compressed, it provides a very weak restoring force. And on average, there's a positive contractile force. So we successfully generate contraction using this asymmetry. And the binding site spacing D is a key parameter determining the magnitude of this force. And here's our results. Here we plot the spontaneous contractile velocity of the substrates as a function of the binding site spacing D. We find a linear relation for large D and a quadratic relation for small d. This quadratic dependence can actually be understood intuitively. Our contraction comes from the asymmetric force extension relation. And in the small extension limit, we can always do a Taylor expansion. Here, the first term is linear, so it's a symmetric term. And the first asymmetric term comes from the second derivative. So we can expect that the resulting contractile force is proportional to the second derivative of tau. And from dimension analysis, we automatically have a squared dependence on D. Also, we find that this contraction becomes considerable when D approaches a length scale delta L, that is the thermal fluctuation length scale of the equilibrium distribution. This is because the delta L is the width of the equilibrium distribution, and D is the distribution of the binding probability. So when D is comparable or larger than delta L, it means the initial Boltzmann distribution is strongly affected by the activity. And in cytoskeleton, the typical values for D is 10 nanometers and delta L is 6 nanometers. So they are pretty comparable in cytoskeleton. Moreover, we can add an external force to try to slow down the contraction. And what we get is the force velocity relation. Interestingly, our result is very similar to that of molecular motors. So this suggests that our mechanism generates motor-like contractility without motors. To conclude, we show that nonlinear elasticity together with non-equilibrium binding-unbinding is sufficient to generate contraction. And both of these properties are generic for living systems. We can test our results using mutated motors with inhibiting motor activities. Or we can also generate contraction on apolar filaments like intermediate filaments and septin, which is impossible for motors. Our mechanism should be considered as an additional mechanism to motors. It also exists in motors and it enhances motor-driven contractility. And we also have results for viscoelastic substrate where we have observed an interplay between the substrate relaxation time versus the binding-unbinding time. Okay, I want to thank to my group members for the help during the preparation of this talk, and thank you for your attention.
All right, thank you, Sihan. So I haven't seen any questions yet, so I have one. Oh, sorry. So we have a question from Bill. Do you want to read it yourself, or do you want me to read it?
Sure, I can read it. Are there candidate molecules that could implement this proposed binding-unbinding mechanism to apolar filaments in real cells?
For apolar filaments, we don't really have any. Now we don't have any like candidates, but as we said, our mechanism is also exists for motors, and we can test our results using mutated motors, so we can design them to block their motor activity while preserving the non-equilibrium binding and unbinding kinetics. So that will work.
All right, so we have another question from Martin. It says, what situations have most likely evolved not to use motor molecules to solve the motion problem, and which ones need motors?
That's a good question. And so I would say that there's a distinct difference between our mechanism and the motors. So that's, for example, here you can see a increase of the contractile velocity as a function of the binding site spacing. And in fact, for motors, we expect the opposite dependence. So this velocity will decrease with D. So for this binding site spacing that is too big, we expect that this non-motor contractility will contribute more than motor activity.
Okay. So I actually have a question about your binding length distribution. You assume that your binding length distributes uniformly on both sides of the origin, right?
Yes.
And then that's why your asymmetric spring give you different forces.
Yes. Yes.
Why can't I think that there's a, when it's unbind, there's actually a uniform, a random force applied on the end, instead of so then this asymmetric spring doesn't give you any benefit, right?
Yes, but as I said, that this PB can be any symmetric function, so it doesn't really need to be a uniform distribution. And
but what if okay, so I can say it's not the so is there any evidence that it's a binding length that's symmetric, not the force, not the random force? Because if so you say it's when it's unbound, the force distribution is uniform.
Yes,
then you already consider the asymmetric spring constant.
So I agree that this in this minimal model, we have used a very rough assumption, a quite rough assumption, but we have also considered a microscopic model. So like this one. So in this one, we really consider a detailed binding potential. This UB here, so it's a periodic triangular potential. And we just let the polymer to bind and unbind on this potential in a way similar to molecular motors. But for molecular motors, this binding potential is asymmetric. And for our mechanism, this binding potential is symmetric. And we can still generate contractility using this detailed microscopic binding unbinding model.
Okay. And is there any experimental measurements on the distribution of the binding length?
I think there are experiments, measurements of this binding potential. So if you know the shape of this binding potential, you will know the spacing between binding sites, and you also know the binding length.
Okay, I see.
I have a related question. Sorry, you might have said this, but like, should it be obvious to me why this binding potential or this binding probability, length probabilities are like completely separate, like uncorrelated to the force that you have at different lengths? Like,
oh yeah, so it's like in our minimum model, the assumption is that the activity is so strong that we can just ignore all the other forces in the system. The binding and unbinding is completely driven by the external energy comes from ATP, so we don't need to consider it. But in our microscopic model, this force or energy of different states are also important.
cool, that makes sense, thanks. Do we have any more questions?
Okay, let's proceed.
Thank you. That was a great talk. That was very interesting. All right, let's move on to our last talk of the session. This will be Shane McNally from Brandeis, who's going to be talking about, well, you can see the title here, Scaling of Subcellular Actin Structures. Thanks.
Okay, great. Yeah, thank you. So yeah, my name's Shane. I'm a joint postdoc with Bruce Good and Yanni Kondiv at Brandeis. And I'm really excited to share some of the work that we've been doing to try to understand how cells can assemble cytoskeletal filaments whose length matches the length of the cell in which they're assembled. And so at the outset of this project, we were interested in trying to answer this very broad question of how do cells control the size of their internal structures. And so for a given subcellular structure, whether that's an organelle or a cytoskeletal filament, for many of these structures, their size has been shown to fall within this sort of natural bounds of they can't be larger than the cell in which they're constructed, but they also have to exist at some size greater than 0 in order to exist. And for many of these structures, they actually exhibit this sort of peak distribution of size where they have some mean size with a narrow distribution. And it's also been shown that for organelles or structures that deviate from this controlled size have improper or inefficient functions for the cell. And so a couple examples that show this, I think, really well are the cytokinetic ring that we just actually heard quite a bit about that you can see here in fission yeast. And so in this case, the length of the cytokinetic ring has to match at least the circumference of the cell in which it's being assembled in order to constrict the membrane of the cells and complete cell division. And another really good example that's particularly relevant in the midst of a pandemic is the ability of differentiated B cells to dramatically increase the size of their endoplasmic reticulum, which is the organelle responsible for secreting antibodies to help fight infections. Our work is focused on cytoskeletal structures, such as those that are formed by actin or microtubules, and mostly because these are really excellent experimental systems to try to understand how size is controlled. Because from a practical standpoint, for the most part, we just need to measure the length of these. Typically, the width of these structures is a fixed quantity. So by measuring the length, you actually get a good readout of the size of these structures. And additionally, many of these cytoskeletal structures are very important for biology. So the mitotic spindle is used for segregating chromosomes during cell division. Cilia and flagella are found throughout all sorts of eukaryotes, including us, but also very cool unicellular organisms such as Giardia that allow them to be motile. Cytoskeletal filaments can also be used to make very specialized structures, such as the stereocilia, which are in your ear and allowing you to hear me speak to you now, as well as actin cables that are found in budding yeast, which allow the cell to do vesicular transport along these cables. It's also interesting that many of the cytoskeletal filaments that compose these structures are actually highly dynamic. So they're constantly undergoing turnover of their monomeric subunits. And you can see that in these sort of cartoon representations of an actin filament and microtubules, where we have some rate of addition R and some rate of subunit removal gamma here. And so when we consider how a cell can actually assemble and maintain these structures at a specific size, we have to think about how these different rates of addition and removal are being tuned by the cell to maintain the length of these structures. And so in budding yeast, which is the organism we're going to be focusing on for this talk today, they can assemble these actin cables, which are you can see them here in this microscopic image and also sort of in the schematic cartoon. So these are linear bundles of actin filaments that are assembled by molecules called formins. And these formins live here at what's called the bud neck of the cell and also at the bud tip. But we're mostly going to be focused on the formins that are here at the bud neck. And if we look at this schematic here, you can see our green formins are able to incorporate these monomeric actin subunits into the plus end or the barbed end of the actin filament. And as they add these subunits, the filament as a whole gets pushed into the cytoplasm of the mother cell. And as more and more of these filaments get assembled and pushed into the cell, they can become cross-linked and bundled to make the filament that we can see over here. And eventually they become targeted for disassembly by a whole slew of disassembly factors that we find in the cytoplasm. And as I briefly mentioned before, these actin cables are thought to be essential for the transport of various cargoes from the mother cell into the growing bud. So in this case, the mother cell is, the cell's undergoing division, so this bud cell is going to become a new mother cell after cytokinesis. And as you can see here, these little red secretory vesicles land on these cables and then undergo transport by molecular motors into the growing bud cell. They also are responsible for the transport of organelles and all sorts of other things. So it's thought that having an intact cable network is essential for the growth and development of the new daughter cell. But what's more pertinent to this talk is it's also thought that the length of these cables is very important, because if the cables become too long, then this transport takes an exceedingly longer period of time. And so in that case, transport would not be a very efficient process. While if these cables are too short, basically it lowers the probability of these sort of secretory vesicles that we're showing here to land on these cables. And they also they don't have access to the cargo that's located toward the rear of the cell. So it's thought that having cables that extend from this bud neck region and terminate somewhere near the rear of the mother cell is an important feature of these structures. But when we look at a population of cells, what we notice is actually there's quite a bit of variability in cell size. And so we really wanted to test this hypothesis that the length of the cable has to match the length of the mother cell for efficient transport. And and so to do this, we went about in a couple ways. We used the natural variation in haploid, which is you can see here in red, and diploid cells in yeast, which have about a twofold change in cell volume. And we also have mutants that we can actually induce about a five-fold change in cell volume by growing them at a restrictive temperature. And you can see over here, this is just an example image of some of the size diversity that we can get with these different cell mutants. And we also found that, probably as you would expect, when you have increased size in volume, you also have increased size in cell length, which is what we define as the distance from the bud neck to the rear of the mother cell here. And so now that we have these mutants in hand, we could go and fluorescently label the actin cytoskeleton in these cells, and then use super resolution microscopy to image these cells and get really nice images that allow us to make quantitative measurements of the length of these cables, again, from the bud neck until they terminate somewhere inside the mother cell. And hopefully you can appreciate here that when you have a larger cell, you also have longer actin cables. But to more carefully make this comparison between the length of the cable and the length of the mother cell, we first just calculated the ratio between these two quantities. And what we found is that for the cells, all different cell types that we looked at, the ratio of cable length to cell length centered right very close to 1, which let us know that, okay, it does seem that the cable length actually matches the mother cell length. But we also noticed is that the distributions of these lengths also seem to collapse into a nice, I guess they all basically look the same. And then we also plotted these cable lengths and mother cell lengths on a double log plot and found that there was a linear relationship between these two quantities. And then we fit these data using the power law to measure the scaling exponent and found that this is very close to 1, indicating that there's an isometric scaling relationship between actin cable length and mother cell length. And so the next thing we wanted to do was actually look at this in real time, look at these in living cells. Because so far we're just using chemically fixed cells where we can just get a snapshot in time of how the actin cytoskeleton looks. And so now we wanted to really look at the dynamics that are underlying the growth of actin cables and how can these actin cables grow to match the length of the mother cell. And so, as I mentioned before, the cables are growing from the spud neck region and into the mother cell here. And if we kind of look at this from a microscopic point of view, we have, again, the addition of actin to the barbed end of our actin filament. And then we also have subunit removal by disassembly factors inside the cell cytoplasm. But when we look at these cells on the microscope, we can't actually see these two different processes. What we really see is the difference between these assembly and disassembly rates at any given time and at any particular length of the cable. And so with that in mind, we went ahead and fluorescently labeled this is a cable marker protein is a good way to think of it, with a fluorescent tag, and then used 3D time series to track the tips of these cables, as you can see here, emerging from the bud neck as they extend into the mother cell compartment. And so now we could actually get real measurements of the extension rates of these cables as for basically their entire lifetime. And so we wanted to think about two possible models that could explain the observations that we have when we look at these extension rates in these cells. And so the first model we considered is a very simple model where we call it the boundary sensing model, where initially the assembly rate is greater than the disassembly rate, which allows this cable to grow from the bud neck into the cell. But then what happens is when this cable essentially just smashes into the back of the cell, there becomes this abrupt shift in these two rates, and the cable extension just stops, and that is what determines your cable extension rate. So the cell doesn't really know how long the cable is, it just knows that it's run into the back of the cell, and it kind of stops extension and reaches its length at that point. And experimentally, the way this would look is if we plot our extension rate as a function of time, you would have a constant rate of extension until the cable runs into the back of the cell, and then it dramatically would just drop off. In contrast, we also thought of something that we call the balance point model. And so in the balance point model, either the assembly rate or the disassembly or both rates are length dependent. And what happens is that these two rates are going to intersect at some length, and that's going to be your steady state or equilibrium length for your actin cable. And that'll determine how long your cable is. Experimentally, the way this would look is, again, if we look at our extension rate as a function of time, cables will grow much faster as they initially emerge from the bud neck, and they will slow down over time here. And so with these two models in mind, we went to our experimental observations and quantified the extension rates in these cells. And what we found is that the extension rate does appear to slow down over time as they extend further into the mother cell and then stop when they reach the rear of the cell. And so this is very consistent with the predictions of our balance point model. And so this is sort of the framework that we're using now to think about how these cables are actually assembled and how their length is controlled in these cells. But ultimately, we want to square these observations with the fact that we see actin cable length is scaling with cell length. And so when we consider these two observations, actin cable length scaling with cell length, and that cables decelerate during extension, and thinking that we have this balance point type model, we can write down this very, very simple equation. This is so much simpler than what everybody else has been showing. It's almost embarrassingly simple, but essentially all this is saying is that our assembly rate K plus is dependent on the length of the cable and the length of the cell. But more specifically, because we see this relationship, and it could also be the same with the disassembly rate. At this point, we're not sure which rate is being controlled or subject to the feedback here in the system. But what we can say now, based on these two observations, is that whichever rate we're looking at, it's actually dependent on the ratio of the length of the cable to the length of the cell. And the important thing that comes out of out of these very simple equations is we can go back and do some more experiments to test these predictions and see if even with this very general framework, if we're close to what we should expect to see. And so using the same, well, and so if we consider that we have cells that have at least a twofold difference in their cell length, what we'd expect is that shorter cells should have a faster deceleration of their extension rate when compared to longer cells. Because if we go back and look here, the length of the cell is going to be much larger in those longer cells. But also if we rescale or normalize our cell length by the length of the cell, we predict that there should actually be a nice overlap between these two profiles. And essentially these initial deceleration rates would be very similar. And so that's exactly what we did. We tagged the same protein that we did on our wild type cells and fluorescently labeled it, and then used 3D time series to track these cables as they extend throughout the cytoplasm of big cells and small cells. And as you can see here, similar to what our model would predict, is that we see faster deceleration from small cells and slower deceleration in large cells. And more importantly, when we normalize that cable length by mother cell length, we actually see pretty nice overlap between these two profiles, at least for about the first half of the cell. There's some deviations here, but we're pretty happy that at least the initial deceleration profiles do seem to collapse here. And so what I've shown you so far is that we do have some scaling behavior between the length of the actin cable and the length of the cell. Actin cables decelerate during their extension from the bud neck and then slow down to the point where they basically stop when they reach the back of the cell. And that this deceleration is a cell length dependent process. But now we really need to kind of fine tune this model some more because, as I said before, we're not really sure which is it. Is it the assembly rate or is it the disassembly rate that seems to be controlling this process? As you can see here in this cartoon model, it could be both. So the big thing we want to do, because we want to get into the molecular mechanisms that are actually controlling this process. And so in my mind, the best step and first step forward is really to try to determine or eliminate one of these rates as being the culprit that is providing this, or should I say is subject to the feedback that is providing this length scaling that we're seeing. And so for the next couple slides and last couple of minutes of the talk, I'm just going to share some very, very recent, like a couple of weeks ago, data that we've collected that I think is starting to help answer this question and help eliminate some of the possibilities that might be out there. And so one thing we noticed just by looking by eyes, we noticed that cables seem to be brighter toward the bud neck region compared to the rear of the cell. And when we did line scans, basically drew a line along the length of the cable and measured the fluorescence intensity, we noticed that it does seem to be brighter toward the bud neck and steadily decay as it reaches toward the back of the cell. And as you can see here, it looks kind of like an exponential, and that's what we fit it to get these decay lengths that you can see here. And so to see if this picture that we drew here, this sort of cartoon, is actually an accurate representation where as these cables are extending, again, because these are bundles of filaments, not single filaments, that these different filaments within the bundle can effectively be pruned by these different disassembly factors. Or I should say that this is consistent with the idea that as these filaments are extending, they're being pruned by different disassembly factors, and that can be why we're seeing this sort of decay here. And so in a way, this can kind of be an assay to look at how is disassembly working in cells of different size. And so it's exactly what we did is we looked at large cells and small cells to see if this cable tapering effect is actually cell length dependent. And so when we look in, again, small and large cells, we actually do see a difference in the tapering. So the large cells have a larger decay length when compared to the smaller cells. But again, more importantly, when we actually compare the relative lengths, we actually see a really nice collapse in these decay lengths, and they look very similar to one another, about half for each of these. So we think this is our first hint into which process to focus on. Instead of focusing on assembly, I think if we were betting, I would say it makes sense to focus on the disassembly rate and start interrogating the different molecules that have been implicated in the disassembly of actin in the yeast cell and start to determine which of those can actually contribute to this length-dependent feedback. And hopefully we'll be able to figure this all out. So that's all I had planned to present. I have to acknowledge people in the good lab as well as the condev group. With 2 labs, there's way too many people to list individually. But in particular, the group that we call the Cable Club with Aldrich, Allison, and Lishi, who's now a professor at RIT. It's a weekly group where we all sit around and talk about all things actin and all things cable related. I also have to thank the NSF for funding to do this work. And with that, I would love any questions. So thank you.
All right, thank you, Chen. Now it's time for questions. Okay, so Josh raised hand. Josh, please.
Hi, Shane. Am I mic on? Can't tell.
Yeah.
Okay. So do you have a candidate mechanism for how the decay molecules know how big the cell is?
Oh, sorry.
You know?
Long story short, no. So we're not exactly sure. So a simple way that this could happen is that some disassembly factor is getting diluted as the cell gets larger. We're not sure which disassembly factor this could be. And there's actually been a lot of recent work done that's showing that different proteins scale differently. I think for many people who've looked at scaling in cells, it's kind of been assumed that the concentration of most molecules stays the same as cell size changes. It's turning out that that necessarily isn't the case. And so I think it's gonna be very useful to dig into these different proteomic and genomic analyses to try to find either assembly or disassembly factors that don't seem to maintain a constant concentration. But beyond that, I think, like I said, it's really tough because it could be anything that's really affecting disassembly that could be doing this. So yeah, it's tough.
Cool, thanks.
Yeah.
Okay. Yeah, you might. Sorry.
To follow up on that question,
Oh.
Like the gradient of the disassembly factor being more concentrated towards the
yeah, that is definitely, and so that's definitely a candidate mechanism that we had thought of. Is if you had a, you know, kind of stealing the ideas from developmental biology with Bitcoin and these things, where you have a gradient that's scale invariant, that would absolutely lead to this. When we've looked, we don't necessarily see a strong gradient in the cells of any disassembly factor. There have been some ideas recently from Jan Bruges' lab, I forget what university he's at, or essentially where they're looking at the scaling of the mitotic spindle. And what they think is that essentially you can sequester molecules to the cell membrane, and that essentially sort of depletes molecules in a cell size dependent way that can give rise to some of this type of scaling. Rebecca Heel's lab has done some similar things with the nucleus, but at this point we don't have any candidate molecules and we haven't seen anything that has that behavior quite yet.
Thanks.
All right. Yeah. So you mentioned that the length of these cables is kind of optimized, right? So and how significant are the disadvantages if they're not optimized for their survival or functions?
Yeah, that's a really good question. So like what happens essentially when you don't have cables that are of the appropriate length, right? Yeah, and so so what we've seen is in mutants with the formins that essentially build these cables, particularly the formin BNI, they actually grow much slower and the cells become much smaller than you would typically expect. So there can be defects to cell growth.
And like, for example, I say all these cables, if they're like 20% shorter, it is serious, or 50% shorter, then you have some real troubles.
Yeah, I mean, I would definitely, again, I don't have data to support this, but I mean, the more severe the defect of the cable, the more severe the defect is going to be on cell growth. I say with something that has 20% difference in length, that's probably beyond the limit of detection for some phenotype that may be happening to the bud cell. But I would agree. So I would anticipate that like severe defects are going to have much more severe consequences on the growth of the daughter cell.
I see. Okay, thank you. Any more questions?
Is there any correlation between the cable length and say the size of the daughter's cell?
So we haven't looked at that specifically. That's actually a very interesting question that I'm starting to look into now, because we've also noticed that as the cells get bigger, you also have more cables that are longer. And so that's exactly what I'm looking at now, is how do defects in the number and the length of cables affect the growth of the daughter? But while I don't think anybody's looked at that yet, what we do see is there is a correlation between the size of the mother cell and the size of the daughter cell. And so there may be some relationship between the cables that sort of feed material to the growing bud. But again, that's a new direction that we're looking at now.
Sure.
Any more questions? Otherwise, let's all thank Shane. That was really interesting. We'll see everybody back at 3 for the next session on physics of behavior.
I'm very happy starting the final section of this symposium. So this is going to be about the physics of behavior, and Rahul Munshi and I will co-chair the session. So again, the framework is going to be similar as in the other session. So 20 minutes talks, 10 minutes questions. You can post the questions in the chat, but we'll also be really happy, if you just raise your hand at the end and and then ask the question yourself, and we'll manage that. And I think sometimes maybe like later on, people might break during the talk, but otherwise maybe post the questions in the chat. And so, so without further ado, I'd like to introduce Bill Gier, I hope I'm pronouncing that correctly, who's doing a PhD at Harvard, and we'll talk about the first beats of a vertebrate heart. So I'm very excited for this talk.
All right. Yeah, so as Marianne said, my name is Bill Jia. I am a student in the labs of Adam Cohen and Sean Megason at Harvard University. And today I'll be telling you about my work characterizing the first beats of a vertebrate heart. Now, the heartbeat is essential for vertebrate life. It's required to pump blood throughout the body, and this permits exchange of oxygen, nutrients, and waste. In a typical human lifetime, the heart will beat about 2.5 billion times, from before birth all the way until death. In the zebrafish, the model organism I work with, the heart's physiology follows a similar trajectory. As early as 24 hours post-fertilization, you're able to see under a light microscope the contractions of the early heart. Now, over the past few decades, we've learned a lot about heart physiology at various stages of animal life. And a lot of this has been aided by the improvement of methodologies with which we look at the system. Here you can see recordings of electrical activity in the larval zebrafish heart through a genetically encoded voltage indicator. Here you can see full volume contractions recorded by light sheet microscopy. And here are some activation maps of electrical activity in the heart when the animal is dying. And our understanding of this has been greatly aided by mathematical modeling, especially of nonlinear and chaotic dynamical systems. We also know a lot about the pathophysiology of the heart, including congenital heart disorders that arise early in the animal's life, and arrhythmias, myocardial infarction, and heart disease that happen as the organ ages. But one thing that we know surprisingly little about is the normal transition from silent to beating in embryonic development. To think about what might happen during this process, we can think about the requirements of a functional heartbeat. So in order for a heart to work, the tissue has to be excitable. So this means that it has to activate in response to electrical stimulus so that it can contract. There needs to be a periodic electrical signal generated somewhere in the heart so that these contractions can happen on a somewhat regular basis. And there needs to be a coherent phase relationship between the cells so that they activate all in one order and blood only flows in one direction. In the adult heart, we know the molecular implementation of all of these processes. Excitability is given by voltage-gated ion channels. Periodicity is conferred by hyperpolarization gated channels and feedbacks and calcium handling in the cell. And phasing requires electrical coupling between cells, which is given by connects and gap junctions. Now, all of these molecules and the functions they confer have to arise at some point in embryonic development as the heart begins to diverge in function from non-cardiac tissues. But they don't have to all happen at the same time. And the ordering in which they happen could lead to really different intermediate dynamics that the embryo has to resolve. So that provides us a natural framework to ask our question, how does the heart go from silent to beating? In the zebrafish embryo, the time period of interest is 18 to 24 hours post-fertilization. During this period, the heart is undergoing dramatic morphogenesis, involving the fusion of two flat sheets of cells and eventual shaping into a linear tube with a lumen by 24 hours. At this point, the heart's already quite similar to how it looks like several hours later when it connects to the vasculature and begins pumping blood through the body. At 24 hours, we have measured in detail the electrophysiological dynamics of the heart, and these can also be easily inferred just by looking at the contractions. There's a relatively constant amplitude and frequency throughout the tissue, and there's a coherent phase relationship between the cells. However, in the intervening period, we know very little about any of these properties. So to enumerate what could possibly happen, we could think about what this looks like on a tissue average scale and what the spatial structure looks like. You could imagine the amplitude of dynamics growing gradually or suddenly. You could imagine frequency doing the same thing. And notably, there could be variability on a shorter time scale before the beat becomes regular. And a phase relationship requires a conduction or wave velocity between the cells. In terms of spatial structure, all of these properties could be homogeneous or heterogeneous. In one limiting case, you could imagine homogeneous frequency and amplitude and a single initiation site, which is the case at 24 hours post-fertilization and indeed in the adult heart. You could also imagine more complex spatial structures involving growing active domains that eventually fuse, regions with different frequencies that eventually synchronize, or multiple initiation sites that fire either synchronously or asynchronously. So now I'm going to tell you what actually happens, starting with the tissue average dynamics. So how we looked at this was by imaging the calcium sensor GCAM7F. So here you're looking at a video of 18 zebrafish embryos from above through a high numerical aperture 2x objective. The raw signal is in yellow and the change in fluorescence is in magenta. You can see in most of the individuals a sort of characteristic patch that shows dynamics that are more frequent and more bright than anything else in the embryo. This turns out to be the heart, and we can extract that signal and analyze it quantitatively. So here are the dynamics for a single embryo sampled at several intervals during this transition period. And you can see at first it's silent, and then the first calcium transients emerge, and eventually these become more frequent and more regular. We can localize the time of the first transient, and this turns out to be about 20 hours post-fertilization. Notably, this is 1 to 2 hours before mechanical contractions are typically reported, which means that we couldn't have looked at these sort of nascent dynamics without a way of visualizing the upstream electrophysiology. We quantified the amplitude of these transients as a function of time, and we see a sudden increase, which suggests that most of the cells are turning on at once. We measure the frequency, seeing a sudden jump to a non-zero value and a gradual increase. And we quantify the variability of the heart through the coefficient of variation of the interspike interval. And this decreases as the heart develops. Notably, near onset, many of the embryos have a CV close to 1, which is like the waiting time distribution for the memoryless Poisson process. This means that there's little to no periodicity at all at the start of the heartbeat. So looking at this data, we immediately saw a qualitative resemblance to a simple model of a noisy relaxation oscillator that's commonly used in neuroscience. Here we have the equation for the model, and we have one variable, our voltage, and 2 parameters, I, resting voltage, and sigma W, a noise power. We also have a threshold, which we arbitrarily set to 1. And once the voltage reaches this threshold, it spikes back down to 0. So what this looks like is that if I is less than 1, we have a decay to a stable equilibrium. If I is above 1, we have oscillations with frequency proportional to I. And if you have noise in the system, then you can get stochastic spikes when I approaches 1, but is still below it. We can quantify the statistical moments of the interspike interval in the simulated dynamics through as a function of I and sigma W, and these indeed match our qualitative intuition about the model. As you sweep across I, as I crosses 1, you have a sudden transition in these properties, and increasing sigma W serves to smooth this out. When sigma W is equal to 0, this is a classic saddle node bifurcation where you have 1 being the saddle and I being the node. When these two annihilate each other, a limit cycle is born. Of course, you can't technically have a limit cycle with 1 variable, but I'm kind of cheating by using this piecewise equation. It turns out that by simple linear scaling of our experimental time to I, we can collapse the data from the previous slide onto a pair of curves for a single value of sigma W. This is clearly not the case for a different type of oscillator model, the Fitzhugh-Nagumo model, which is an example of a different type of bifurcation. So what this means is that a very simple model describing a bifurcation of a single oscillator does a really good job at describing the sort of transitioning dynamics of the heartbeat. This is actually kind of surprising because, as we know, the heart is made up of many cells, and it turns out that embryonic cardiomyocytes mostly will have spontaneous oscillations if you dissociate them. This means that one might naively expect that when these dynamics emerge, you have independent oscillators that eventually couple. That is the case in many biological systems, but apparently not this one. To understand why this model is doing so well, we decided to perturb the system using genetically encoded optical actuators and sensors. Specifically, we expressed the orange light excited calcium sensor, FR-gecko-1c, along with a channel rhodopsin, a non-specific cation channel that is opened by blue light. So what should happen here is when the blue light stimulates the sample, the cell membrane should depolarize. That should result in opening of voltage-gated calcium channels, causing the intracellular calcium level to rise, and then that should be read out as an increase in fluorescence in this sensor. Because these two engineered proteins are spectrally orthogonal, we can directly compare the baseline and transiently perturbed states. We have a custom optical setup to pattern these light sources in time and space programmatically. And this gives us exquisite spatiotemporal control over the electrophysiology of the heart. Here's the system in action. So the orange trace is the change in fluorescence of the FR-gecko-1c. And you can see that in the first 20 seconds of this recording, you see the endogenous dynamics of the heart. And then once we apply blue light, we can pace the heart much more quickly than its sort of own activity. We can pattern this perturbation in space. So here you can see the wave propagation of the endogenous activity. And when we deliver stimulus to this yellow spot, you can see propagation emanating from this source. And these are clearly not in the same direction. So what this means is that we can essentially overwrite the periodicity of the heart and the phase relationship between the cells. An important implication of how these tools work is that since we're able to transiently perturb the system, we can actually assess the presence of electrophysiological properties before there's any endogenous activity to measure. So here we have the system looking at an embryo before it's showing any endogenous activity, and then after. So on the left, you can see the baseline where it's silent at first and then shows activity. But when you apply stimulus, it's responsive even when its endogenous activity is silent. This means that the tissue is excitable before it is spontaneous. When we pattern the stimulus in space, again, in the sort of silent tissue, you can see that some of the tissue is able to support wave propagation. So what this means is that the heart is basically ready to beat, and it can beat if you give it external instructions. It's just waiting for some spontaneous activity to emerge. So an important prediction of our model in this process of transition is that the heart should become increasingly sensitive to external stimulus as it approaches bifurcation. So here we have the model with different values of I, and we're applying external stimulus to imitate the channel rhodopsin. And as you can see, the stimulus more probably elicits a spike as I increases. When we perform experiments on a single embryo several times over about 20 minutes of development, we see something encouragingly similar. So I've told you about the amplitude and frequency characteristics of the heart as it's sort of developing, and suggested that these should be spatially homogeneous and that a single oscillator explains all of this. But what about the phase relationship? To characterize this, we performed high-speed imaging of GCAMP7F across various points of development. So here we made isochronal maps from several spikes averaged at different time points for the same embryo over 2 hours. Here, the first one is less than 30 minutes after the heart has started beating. You can see that there's a single locus of initiation and that it stays relatively constant in space as the embryo is developing, even as it starts to undergo tube morphogenesis. It turns out that for most of the embryos that we looked at, this was the case. And intriguingly, this locus sort of localizes to the left side. So the sum of the past two slides basically tells us that it's likely that spontaneous activity emerging at this locus of initiation is what finally drives the start of the heartbeat. In other words, this locus is probably that single oscillator that we're looking at. So what I've told you today is that we've mapped the earliest electrical activity of the zebrafish heart, which occurs before contractions are visible. The amplitude of this activity increases suddenly at its onset and remains relatively constant, and phasing from a single locus of initiation is established early on. These observations and the evolution of the frequency, mean, and variability are well described by the saddle node bifurcation of a single noisy relaxation oscillator. The spatiotemporal structure described is plausible because many cells are already excitable and support wave propagation. Therefore, the emergence of spontaneous activity at a single point should be sufficient for phase coherent activity throughout the bulk of the tissue. What we're interested in looking at next is what's special about the cells at the locus of initiation. So what I told you before about this sort of leftward bias and then and then the fact that all the most cardiomyocytes are spontaneous suggests that there must be some sort of biological factor determining where this locus is. We're also interested in looking at whether electrical dynamics affect cardiac developmental progression, because this activity turns on really early in development. And you could imagine various feedbacks that refine it into its adult state. With our longitudinal imaging and channel rhodopsin perturbation, we're well set up to answer that question. So I'd like to thank my co-advisors, Adam Cohen and Sean Megason, for their tireless support, and all the members of both of my labs for creating stimulating and diverse working environments. I'd like to specifically highlight the contributions of Yutong Qi and David Wang Campos in the Cohen lab, who were instrumental in engineering the optical setups required for my experiments, and Natasha Brown in the Megason lab, who helped me keep some of them going when the timings were inconvenient. Thank you very much, and I'm happy to take any questions.
Thank you, Bill. That was an excellent talk. I don't see any raised hands yet, so I can probably start off with a question. So yeah, you mentioned that, I mean, you would be working next on finding out what's the starting node actually all about. But do you have actually a clue of how the gap junctions might be propagating from whatever the starting node is, the signal that is?
Yeah, so based on my experiments, based on this experiment over here, it suggests that the gap junctions are already kind of in place when the spontaneous activity turns on. We're looking at ways at various schemes to try to measure the formation of these gap junctions more precisely, and the zebrafish is a really powerful system to do that. But this is all I have right now.
Perfect. Bill has a question, and after that, I'll ask you another question if we don't have any more from anybody else. Bill, please go ahead.
Yeah, thanks. That was a lovely talk. I mean, maybe part of this is answered by the measurements of variance, but as you approach the bifurcation point in the presence of noise, it's not only that the curve particular functions should have universal forms, but sort of distributions of things should also be predictable, right? So if you, for example, if you try to sit right at the bifurcation and measure, the distribution of beat amplitudes or frequencies or something like that, can you make progress in that direction? I know it's much harder to measure a distribution than a moment, but.
Yeah, experimentally, I'm not sure. I'm not quite sure like how we would be able to get the embryo to sit at that bifurcation point for long enough to get enough statistics, but sort of trying to take a small window, I have measured the distribution of the interspike intervals, and it sort of looks encouragingly like an exponential, with obviously with a lower cutoff due to the absolute refractory period of the cells.
Mm-hmm. So Bill, if you optogenetically hyperpolarize again a bunch of cells and keep an equilibrium, won't that be able to keep your things at the bifurcation state for long?
Yeah, that's a possibility. That's something that we're looking at, the sort of shifting of the. So if this is the real mechanism and V is our actual resting potential, then we should be able to move it up and down with sub-threshold stimulation of either the hyperpolarizing or depolarizing channel rhodopsins. So that's something that I'm also working on.
Perfect. Marianne has a question.
Thank you. Thanks for a great talk. So I was actually really impressed by the fact that you showed that the noisy relaxation oscillator was able to reproduce your behavior while the Fitzhugh-Nagumo model was not. I think that's really nice. So I was wondering, like, do you, in sort of similar in the vein of the question that Bill asked, do you see when you look at the populations of your embryo that there are also some that set on without the threshold? And can you fit an eye in the sigma that sort of reproduces your population statistics?
Wait, so sorry, what was the first part? Something about
so you've shown this. yeah, you've shown this like slight threshold with the onset of the amplitude, right? That there was a little jump and then it goes up.
Yeah.
Are there some of them where it's smooth? So do you see no thresholds at all? Because in your fit, you saw that some eyes and signals, I think, gave you a smooth.
So in terms of the amplitude, we didn't see any that were smooth. The sort of increase after the onset, there's part of it that's due to that sort of fitting of where 0 is being a little bit imprecise. There's part of it due to like focal drift of the microscope, and then there's probably something to do with like calcium handling in the biology that's like not like directly related to our model. Yeah.
I see. Okay, but that should narrow down your parameter choices, I guess, then. Okay, great.
We have 2 questions in the chat. Sorry, Marianne, did I cut you off?
Yeah, no, perfect, thanks.
Oh,
Thanks.
okay. We have 2 questions in the chat. First is from Antonio. Can you actually read, or I can read it out for you? I'll still read out the first question. Really cool talk. Naive question. Do you know what portion of cells have spontaneous oscillations? If all cells have spontaneous oscillations and they're coupled, what's the mechanism that avoids synchronization and allows for propagating waves?
So the first part of this question, in zebrafish, we don't know, and I'm probably going to do that experiment at some point. But in the chick, it's at corresponding stages, it's about 70 to 90% of cells have spontaneous oscillations. And in terms of the mechanism that avoids synchronization and allows for propagating waves, that we think is overdrive suppression, where essentially the fastest cell will determine the frequency of the whole tissue. And that's potentially why we see this single oscillator, even though other cells have spontaneous capacity. It could be that it's just getting a head start over the other cells, or it's always just like faster somehow.
So when you say 70% of cells, you mean 70% of cardiomyocyte cells?
Yes, 70% of cardiomyocytes, sorry, not 70% of all cells in the embryo.
Okay, so there might be some other mechanical, mechanically influencing cells, which probably propagates the direction of waves through, some mechanical stress, because there are also mechanical stress sensors on the membrane, right?
So in my case, like we've disabled mechanical contractions by blocking troponin, so that optimizes from contracting.
Perfect. Yeah, we have another quick question. This is from Nirag, who's our next speaker, by the way. So the question is that, is it a fundamental feature that the topology is saddle node, or are the observations consistent with other types of bifurcation?
So this is I'm a little bit new to this area of physics. But I believe that in 2 dimensions, there's only really the saddle node bifurcations and like 2 different types of Hopf bifurcations that can occur. I'm not sure about higher dimensions, but I basically ruled out like the lower dimension models.
Fair enough. So I think we are in time. If we don't have any other question, I think we are in time to move on to our next speaker. So next speaker is Nirag Kakaria. He's a postdoc from Yale. And the stage is yours, Nirag. And one second. So Nirag actually mentioned that he would welcome any questions in the middle of his talk, right?
Yeah, there's a couple moments where I'll just make sure that the understanding is clear, because I know maybe like kind of the backgrounds of the audience is pretty diverse, but I'll keep it under 30 minutes.
Yeah, perfect. Thanks.
Okay, thanks. I just want to mention I've been my daughter's daycare closed all week, so I've been running around her. And my wife just walked in the door like 5 minutes ago, so I'm a little frazzled mentally, but I think it'll be okay. And she's got the baby right now, so I think we're fine. I'm going to talk about this work that we put in the archive last week, in which we've identified a novel stream of information that we found to be critical in in odor navigation in complex environments. So let me just start with an example of odor navigation in such an environment. And this was taken by my colleague Mahmoud, who designed this beautiful experimental paradigm. What we have here are flies that are walking in this large assay in 2 dimensions. And his breakthrough is really identifying an odor that could be seen at the same time that we can measure the animal behavior. These experiments are done in the dark, so the flies can't see the odor, but we're imaging in the infrared, so we can essentially map both the signal and the animal behavior at the same time during these experiments. You can notice that in general, the navigation strategy is, the flies have a little bit of trouble getting to the source, and that may not be surprising if you look at the odor signal, which is shown here in the bottom right. It doesn't have a smooth structure and it lacks a lot of regularity. Instead, what you find are these bouts of high concentration interspersed with these long periods of clean air. So this very intermittent nature of these type of odor environments has inspired a lot of modeling studies and a lot of our understanding, which is that navigation in these type of environments is mainly driven by the temporal features of the signal, such as their timing and their frequency, with less emphasis on the spatial changes in concentration. But if we look at the fly anatomy, there is something very suggestive about it, which is that flies detect odors using these 2 cylindrical-like appendages, which are their antennae on their head. These are spaced by a few hundred micrometers. And it seems likely that if they could detect concentration differences between these 2 locations, they could use some sort of gradient ascent or some sort of gradient measurements to get to the source. In some situations, this is the case. It's been shown that in this type of situation where you kind of tether a fly, you hold it there, but it can still kind of move left and right. And they have an air stream coming towards a fly that's laced with an odor. When they just have the air, the fly's orientations are kind of angularly dispersed. When they add the odor coming in, it tends to stay in the ribbon region by moving back into it. However, when they make it such that one of the antennae can't sense odors, this ability to stay in the odor ribbon is severely degraded. This really suggests that they're using some sort of interantennal concentration difference comparison to stay in the odor, essentially by computing gradients. What we're proposing in this work is that in dynamic environments where gradients are much less indicative of the source, this type of bilateral sensing is used for a pretty different computation, which we're claiming is detecting the motion of the odor signals more than the linear gradients, more than the static gradients. So the first thing we have to do to experimentally test this is disassociate the odor from the wind, because obviously odors are natural, plumes are carried by the wind, so their motion will be correlated. So to do this, we turn to an optogenetic approach, which is the same thing that Bill mentioned in his talk. Essentially, we express photoreceptive proteins in the olfactory sensing neurons, which allows us to stimulate the neurons using light as opposed to odor. Our assay essentially is just a 2-dimensional arena where flies can walk, and we designed this setup where we have a movie projector mounted on top, and the movie projector basically just projects these virtual movie environments that are virtual odors to the fly. Then the flies are blind, so the only signal they have is this fake odor cue. Here's just an example of that in real time. So you kind of see that we're just tracking the fly and then presenting it this fictive odor. And the flies are blind, so they can't really see the red, but they think that they're smelling something when they get hit with the red light. I'll take any questions now because this may be a different type of setup. So if there's any questions on how this is working or what we can do, maybe you can just say them verbally.
I see no questions so far.
Okay, yeah, that's fine. Feel free to interrupt if you'd like to. So what we first did was, again, our goal is to see if flies can detect the motion of odor signals, so the direction that the signals are moving. So what we first did was we presented these bars moving either left or right at a constant speed. And what we found is that when the bars move to the right, I know this doesn't render very well on Zoom, but when the bars move to the right, the flies tend to bias their heading and direction into the leftward side of the arena. This is pretty exciting, and I'll emphasize that this is really the only directional cue that the flies have. There's no wind here, and the flies are blind, so there's no visual inputs. So it suggests that they can really sense the direction of the odors in absence of any other cues. And we kind of wanted to look into this in a little bit more detail. So we then used much wider fictive odor bars, so we can basically test their behaviors at the on edge when the bar first passes into the fly, and the off edge when it passes over it. And this in particular allows us to disassociate gradient sensing from direction sensing. because at these edges, the gradients for the on edge would point to the left here, whereas for the right, it would point to the right. So you'd expect to see different behaviors at these two edges, whereas the direction is well defined in the rightward direction. So you'd expect to see similar behaviors there. In particular, if we plot the turning direction as a function of the fly's orientation, we'd expect to see something that has peaks when the flies are parallel oriented to the edge, but the peaks would be asymmetrical for gradient sensing, and they'd be in the same direction for direction sensing. The observations are completely consistent with direction sensing. So at the on edge, they turn against the direction of the motion of the bar. At the off edge, they do the same thing, turn against the motion of direction. And this trend goes away when we have flies of the same genotype, but where the optogenetic activation is not activated. Now, I mentioned that this is somehow related to the fact that flies have two antennas. So how are they computing the direction of motion of odor signals? And the idea is that we're basically borrowing the ideas from motion detection in the visual circuits. This has been studied in detail for several decades, and there are still many models that can explain observations of how motion is detected, but actually a lot of them that are consistent with the observations share some particular features. And these generic features are that you have to have two spatially separated sensors that respond to different parts of the physical space. So in the eye, this would be like within one eye, there's these, you can kind of see there's these photoreceptor regions, and the neighboring ones respond to different parts of the visual space. The other feature is that one of those has to have some sort of asymmetry where the signal is delayed as it goes downstream, and then these signals are multiplied. The idea is that when there's an object moving from left to right, it'll hit the first sensor, and that sensor will detect it. They'll have some signal going downstream, but it'll be delayed. And by the time the object gets to the rightward sensor, they'll be coincident on the part that is multiplying those signals. So you can get a detection of motion in the rightward direction. How the delay is implemented is often modeled as a linear filter, which can have various forms. For example, it can have a single hump that's away from t equals 0, but there's other ways to model it. The fully direction selective correlator, this is called the Hassenstein-Reichardt correlator, by the way, has the same architecture, but it's added to its reverse, and the reverse side has a negative sign. So what this detector would do, it would measure direction signals going from left to right with a positive sign, those going from right to left with a negative sign. So it's fully directionally selective. Our hypothesis is this is happening essentially in the fly's olfactory circuit, where these two sensors are literally the two antennae, and these signals are being asymmetrized down in second and third order brain regions, and then being computed downstream to calculate the direction and motion of odor signals. Assuming this model, we can actually use our data that I showed a moment ago to extract some of the features of how these delays are being implemented. And so what we can do is first just get a prediction for what the response would be to a moving edge. And it's very simple. Essentially, the moving edge, when it hits the first sensor, it's a step function that's linearly filtered, which gives this portion. And then it hits the second sensor with a delay of delta t a little bit later, but that's not filtered. So you just multiply those. The reverse would be a similar thing happening, but coming from this arm. And then so we're assuming that the turning bias essentially takes the output of this correlation algorithm and then integrates it over some short amount of time. And it gets this very simple form, which essentially allows us to relate the behavior of the fly to the delay it takes for the signal to hit the two sensors, the two antennae. And from that, we can extract the time scale of this delay that's computing this. The delay between the two antennae can be inferred from our experiments just using some simple geometric considerations if we know how fast the fly is walking and how fast the bar is moving. And so that we can express the delay just in terms of geometrical quantities. And then we can just plot the behavior as a function of this prediction, from which we can extract a time scale, a filtering time scale of somewhere between 10 and 50 milliseconds. This is a pretty rough estimate, but it is not too different than what happens in the visual circuit, where it's also on the order of tens of milliseconds. And it also suggests that the speeds that this correlator is sensitive to would require this type of delay between the two antennae, which basically corresponds to odor signals that are moving somewhere in the range of 10 to 100 millimeter per second, which is not too far off from wind speeds that they would normally encounter in the wild. Now, the next thing we wanted to do is we wanted to understand a little bit more about the predictions of the Reichardt correlator that I mentioned a moment ago. Fundamentally, what those correlator algorithms are doing is that they're calculating correlations in space and time. So what I'm showing you here is an example of a stimulus that has well-defined correlations, but even if it doesn't have a coherent object moving in one direction. Again, I know it doesn't render very well on Zoom, but you may be able to see that these bars look as if they're kind of traveling in the rightward direction. And that motion percept is simply related to the fact that the bars at each time, that's in neighboring pixels at neighboring times, are correlated. So here's an example of a snapshot of these bars at 3 different times. You can kind of see there's like kind of a bit of a downward skew because the bars are correlated with neighboring ones in the rightward direction. So the experiments we can set up are essentially what we're going to do is we're going to flow wind in one direction. So now we're going to start adding wind to this. But we're going to have correlated stimuli that looks just like these bars, and the correlations either go with the wind or against the wind. And we want to see if flies can pick up this percept. The correlations, these stimuli we're able to design stimuli using a previous approach that was mentioned in previous papers, where you can get a stimulus that only has correlations with neighboring pixels at neighboring times. So this correlation matrix essentially says that one frame later, this pixel is correlated with its neighbor, but it has no other correlations anywhere in the arena. We can correlate in the opposite direction, which basically means that these bars appear to be moving in the left. Now, there's something else we can do, is we can actually make the correlations negative. And what that means is that there's a bar at this pixel. It's likely to be followed by the opposite color bar at the neighboring pixel in that direction. All right, so they have, let me be very clear here. The direction is still well defined, but positive means that red is followed by red, whereas negative means red is followed by black. So the contrast inverts. Now, why would we do this contrast inverting correlation? And the reason is because the output of a Reichardt correlator or similar type of algorithms depends both on the correlation direction and its polarity, and it's a product. So you can actually reverse the percept by flipping the correlation sign. Now, what this means in practice is that and normally I could show this in if it was a live talk, we can actually show the negative correlations. And what happens is that if something is moving, if it has a correlation displacing to the right with a positive correlation, we would perceive it as moving to the right. If it has a correlation displacing to the right with a negative correlation, we actually perceive motion in the opposite direction. You can actually see this, and I'll show you a link on the next slide, and we can look at that online. So the idea is that if this correlation algorithm is being used by flies for odor motion sensing, then these percepts should flip when you flip the correlation sign. The precision of our experimental assay allows us to test this because we can actually stimulate individual antennae because it's very spatially resolved. And so we can give them these type of correlation stimuli. So just to look at this real quick, when we have positive correlations, when the correlation's in the same direction as the wind, we expect the flies to turn against it, just like they did with the bars, which they do. When we misalign the correlation displacement direction of the wind, the responses are heavily suppressed. And we see that these responses are completely reversed when we change the correlation sign to a negative one. So this one here, the correlations are displaced to the rightward direction, but the signs are actually reversed, and it actually makes the flies have a similar percept to when it was in the opposite direction here. So this was very exciting because it's basically saying that motion detection can be enacted in 2 very distinct sensory modalities using a computation that's consistent between them. This doesn't say anything about the anatomy, so whenever I present this to true neurobiologists, we have to be very careful about saying that we don't know that there's 2 detectors, and if they're using some sort of filter, it may be much more complicated, and I'm sure it's much more complicated. But fundamentally, there has to be some sort of correlation algorithm happening. This is really just a motion illusion, the one that it flips, and you can take a look at an example of this online at Damon's website. Okay, so if flies can detect the motion of odors independent of the wind, if they use a correlation-based algorithm to do so, what's the use of this in actual navigation? So the first thing we wanted to do is understand, well, does the motion of odors present or provide an independent cue from the wind, or is the motion of the odors always the same as the wind? Is it just a redundant cue that may be able to reduce or to increase SNR, but it doesn't give anything more than that? We just first started with some simulations of odor plumes, where this simulation can give us something the experiment can't, which is the wind direction. Overall, in general, here we have kind of a laminar flow this way, but it's perturbed, so it ends up creating a more complex flow, and it makes the plume break up a little bit. In general, the wind direction is mostly downwind, and I'm only showing the wind direction where the odor plume kind of exists, so mostly it's downwind, but you'll find that the odor motion, which I found by taking correlations of the simulations across frames, the odor motion has this very strong component coming laterally outward from the plume axis. This is a very suggestive type of directional cue, because what we know from olfactory navigation in insects is that in general, when they get an odor signal, they tend to turn upwind. There's a century of studies going back indicating this. But this provides an orthogonal cue, because now instead of just going straight upwind and then getting to the edge here and then self-correcting and going back, they can actually follow it more directly to the plume source. So it allows them to localize the plume in both the upwind direction and the lateral direction. So this could enhance navigation and may be relevant for natural plume navigation. One thing we did in addition to this is we also investigated this outward lateral motion with just some very simple models of turbulence. So we're using a very simplified model of odor signals in a turbulent plume. And these odor signals are the odor is really just modeled as a series of packets that are released from a center, from a plume central location. The packets grow with molecular diffusion as they go downwind, and they also are infected downwind with a constant speed. And they also have a diffusion outward, which is given by what's known as a turbulent diffusivity. So that diffusion constant in the lateral direction is really dictated by shear forces and the presence of eddies. That diffusion is much larger than the molecular diffusion. So it makes this spread in the lateral direction much larger than the spread of the individual packets. And this kind of illustrates that. And so what we're going to do is we're going to use this simple model to just understand what is the average motion of a packet at a given region, particularly in the lateral direction. And does that average motion have a well-defined direction away from the central axis? We just use a model that was, or an understanding, I guess, that was really suggested a century ago by Taylor, in which he essentially modeled not the packets as a diffusive process, but he modeled the speeds by an OU process, by an Ornstein-Uhlenbeck process. So the speeds themselves are following an OU process, and this ends up giving a correlated random walk. The correlated random walk over longer times essentially resembles diffusion, but on short times, the things are correlated. What we're going to do in our modeling is just approximate this by a telegraph process because it makes some of the calculations simpler to deal with, but that's okay because actually their time correlations are the same in the sense that the correlation between the velocity decays exponentially in time with the time scale big T. The question is, what is the average packet velocity at a fixed location from the plume axis? And we can just kind of intuit that by basically saying, well, the packet velocity at a given location is, because the packets in this telegraph process are moving either rightward or leftward with the same velocity, it's the number of packets moving to the right minus the number of packets moving to the left. So we just take the number of packets moving to the right, which is essentially the number of packets at that time, multiplied by the number of packets that got there, which is exponentially suppressed, minus the number of packets to the other side, times the number of packets that got to that location, which is also exponentially depressed. Now, the reason there is an asymmetry here is because there's more packets to the right, so the number of them going in the positive direction would be higher than those going in the leftward direction. By approximating this by a telegraph process, we can just get a closed form solution for what that looks like. And I'm not going to put the formula here, but essentially what you find is that the lateral velocity has this nice asymmetry where it essentially points outward from the plume axis. Another key point is that the asymmetry depends on the correlation time. The correlation time increases, the asymmetry becomes stronger. So it's really fundamental that we're modeling this not by direct diffusion, but by a correlated random walk. Okay, so that indicates that in these very simple models of turbulence, there is generally an outward direction of the odor motion at any given point. And the next thing we can do is, well, we can ask ourselves, what would a 2-point sensor measure? Can it pick up on that? And this 2-point sensor is basically modeled on the Reichardt correlator I mentioned earlier. And I just modeled this as a very simple way to compare concentrations at two locations. So basically, I'm defining this quantity that says the concentration at one location times the concentration at a neighboring location, but the time before, minus the same thing, but time antisymmetrized. So basically this compares concentrations at neighboring locations at a different time. In a small limit, this basically ends up having a couple of terms, one of which really picks up the velocity of the packets, and then one which depends more on the position of the packets. by taking an average over this over all the different packet trajectories, we find that there is a well-defined motion direction that can be picked up by this correlator that does point away from the plume axis. One thing that's interesting about this is that there is a trade-off, or there's, I guess, a regime in which the velocity, the correlated velocity matters. And so when you look at the velocity of the packets as a function of the molecular diffusion, if the velocity is too low, in other words, this is a kind of approaching the limit where molecular diffusion is taking over the correlated random walk of the packets, then the asymmetry of this starts to reduce. So it really depends fundamentally on the fact that these have some small correlation in time. So we can test that. This is the last thing I'm going to show is we can test these predictions by essentially using our assay in which we can present plumes where the odor motion is, these are virtual plumes. We're making the odor motion have this outward component, but we can also reverse it artificially. And you'll see that when the motion in the lateral direction is reversed artificially, the flies can still get upwind towards the source, but when they get to the plume edges, they have a lot of trouble maintaining within the plume cone. So much so that the ability to get to the source region is decreased by more than 50%. So this lateral motion really does drive their navigation towards the source. The last thing we can do is a very elegant experiment that was suggested by Thierry and Damon, which is that you can actually present a natural plume and you can keep every aspect of it locally the same, but you can reverse the odor velocity simply by playing the plume backwards. This is something that we can do with our virtual plumes. So if we just do this, essentially just playing the plume backwards, we find the same thing. With a reverse plume, in which the gradients are preserved, the spatial distribution of hits are preserved, but the velocity is reversed, has a huge degradation in the ability of the flies to get to the source. So what I've shown here is that the first demonstration that animals can sense the direction of motion of odors and that they use this using an algorithm that is consistent with visual motion detection. I've also shown that odor motion seems to be a generic feature of naturalistic odor plumes, and the flies actually use this to get to the source in a naturalistic navigation task. I'd like to thank my main PI, Thierry Monet, also Damon, they were basically equal in designing the project and then the funding sources, and I'll be happy to take any questions.
Thanks so much for a super exciting talk. So yeah, well, maybe I don't see any questions in the chat right now, so maybe I'll start. So actually this is maybe a small hobby question of mine, but can you vary the intensity or is this an on-off process? Like in your stimulation, I mean, is there a way to simulate intensities?
Yeah, absolutely. If I go, well, I don't need to go back, but basically we're just using a movie projector. So the movie projector has, you can change the intensity. It's basically an 8-bit integer. So you can change the intensity. Now, what we could do, we actually did this recently, is we could you could find a way to somehow map the intensity of the virtual odor to an actual odor concentration. And then once you get that mapping, you know that if I make the intensity half as much, the odor is whatever. There's some mapping between them. And then with that, you could vary the intensity and investigate those. And that's a very important point because we're treating everything really here as binary. That's a huge approximation. And it's also a huge approximation of the fact that the temporal dynamics of this are just dictated by on and off switches, but odor signals in nature are much more complex. So the intensity is something we are looking into, and it's a great point, but that does require a little bit of engineering. And it's, when you talk to neurobiologists, like we're using channel rhodopsins, which are ways to stimulate the neuron using light. They don't really approximate the kinetics of the neurons very well. So there's a lot that's lost by doing that. So the question of whether this is a quote unquote real odor percept is something that we don't really know. We just know that they're attracted to it. So that's good enough.
That's great. That's great. And can I ask a quick follow-up just because there was something that I didn't quite understand. So you said it was very important to show that the random walk was correlated, and that was because the velocity then was asymmetric. But so it asymmetric in the mean you meant that it went out of the plume, but was there also like an asymmetry in this direction? Like I didn't quite understand.
No, I'm only considering in the lateral direction. So in terms of having the downwind component to the velocity is dictated by the mean flow. The lateral components in the odor motion depend on this a little, they depend on the correlations in this random walk. Now that's just for the odor motion. The wind motion at any given point, the wind will average out to 0. So the key point is that the wind fluctuates, but it has no overall mean. But those fluctuations starting from a point source will eventually make a net flux of odor motion outward. So that's the key point, is that the odor motion has a well-defined direction, even though the wind doesn't in the lateral direction.
Okay, great. Thanks very much. Very exciting. I see that Rahul has a question, so maybe I can.
Yeah, I had a very naive question basically. So if you kind of introduce your order mimic, which is the light, right? Continuously. I mean, no pattern, no motion, nothing, just a.
So just the entire arena uniform
Yeah,
light.
entire arena. I mean, what would you expect the fly to react like? I mean, that might be one of the limiting cases of your model.
Yeah, that's something I probably should have given a little more background about. When you give just an odor signal to the fly, and it doesn't, let's say it has no gradient, it has no direction, whatever, just an odor signal on both antenna. When it can sense the wind, the fly will go against the wind. This is well known, and we've tested this in our assay. It's well known from real odors. So the base strategy, the basic strategy is just I sense an odor and I go upwind. So that's what we'd expect if it's just uniform. I also did experiments where, and I didn't show them here, where I had bars moving in random directions compared to the wind. And what we found is that the behaviors in response to both the wind direction and the odor direction seem to be summed, just simply summed. So they just take the vector of odor motion, vector of wind direction, and then sum those vectors and then turn against that direction. I'm not sure if that answered your question.
Yeah, I guess kind of. I mean, I was also imagining the situation where the wind would not be involved at all. So.
Oh, just odor by itself?
Yeah.
Yeah, so if it's just odor, so this is something that really requires a kind of a virtual environment, because you can't really have an odor by itself unless it's like a patch and maybe just diffusing outward. But let's say we did it in our virtual environment where we had just uniform odor, virtual odor everywhere. In general, the flies will move around more, but they won't have any they won't go in any particular direction. If you have it in a patch, then they get to the edges, they'll realize, oh, I missed where the odor was, so I'm gonna go back into patch. So there is some foraging aspect to that. But the directional, there's no bias direction in that sense.
Okay, very cool. I still don't see any questions in the chat, so maybe, I guess, probably wise to move on, but thanks very much for a very interesting talk. This was really exciting. Thank you. Great, so then I'll introduce the last speaker of the session, Bashi Zhong, who's doing a PhD at Georgia Tech, and he'll talk to us about lizards, in particular coordinating tiny limbs and long bodies. So looking forward.
So hello everyone. Today my topic is coordinating tiny limbs and long bodies, geometric mechanics of diverse undulatory lizard locomotion. So before we start, let's take a look at some biomechanics and analysis, how those analyses can help us understand the relationship between the body forms and the locomotive function. So here we show two types of snakes, the sidewinder and the connectors. The emergent locomotion patterns for connectors is a slithering, or for sidewinders is a sidewinding. So it's totally distinct different directions. So accompanied by the difference in locomotion patterns, those snakes have different skin structures. The AFM scan reveals that the microscopic structure for connectors is anisotropic, and for sidewinders is isotropic. In this way, how and why should the locomotion changes be associated with the changes in the skin structure? So that becomes a question. So, and biomechanical analysis can provide insights in evolutionary convergence. Specifically, the isotropic skin structure can be beneficial for sidewinding in going sideways, and the drug anisotropy can help the connectors better slithering. So let's take another example. So in the evolutionary transition from aquatic life to terrestrial environments, it requires modification of several body systems. And biomechanical analysis showed that the tail use improves the performance of vertebrate land locomotors on soft substrates. In the course of evolution, many species become extinct. While we can still infer the morphology from the fossil studies, there is missing information on their locomotive information. Biomechanic modeling can reverse engineer those locomotion patterns from fossil tracks, giving us information on their locomotion evolutionary pathway. So those insights can help us better understand the selective pressure from a locomotive perspective. So here, we use biomechanical models to study the limblessness in squamate evolution. In squamates, which is lizard and snakes, the snake-like body forms have been evolved at least 25 times. The evolution of snake-like form involves the elongation of the body and the loss and reduction of the size of the digits. While the exact select pressure is unknown, the elongate morphologists hypothesized to aid locomotion in fossorial and clustered environments. However, the mechanism of proportion in such forms, for example, the use of body and the use of tail to interact with the substrate, and the potential body-limb coordination remains unstudied. For example, the body movements in lizards and snakes are distinct from each other. Specifically, lizards, as we all know, use standing wave to body bending to assist leg retraction protraction, and snakes use a traveling wave to generate thrust. How about the intermediate species between lizard and snakes? How should those intermediate, which is short limb, elongate, skinks, use their body? So their existence of multiple extant intermediate species in the transition between the quadrupedal and snake-like is critical to investigate locomotion evolutionary pathway. A few clades satisfy this criterion. A Filipino skinks, which is Bertamini, is one of them. Thanks to Professor Bergman, here we have a video recordings of locomotion patterns for different skinks. Here's the Bertamini monicame, and this is Bertamini taylori, and we will see Bertamini catwa here. So those are the intermediate species, short-limbed and the elongate forms. So we compare 7 species such that they form a spectrum of body elongation and limb reduction. L, which is the relative limb size, range all the way from 0.33 in fully limbed lizards to 0 in snakes. Qualitatively, we observe that the node, which is the body part which has 0 curvature, is stable in fully limbed lizard, one at the shoulder, one at the hip. And in the snakes, it's propagating from head to tail throughout the gait. But for those intermediate species, one of the nodes is stable near the shoulder, and the other node propagates from the mid-body to the tail. So now we wish to quantify those observations. So to quantify those observations, we measure the body posture by body curvature profile, which contains the spatial temporal information of the body curvature. Prior work suggested that the essence of lateral undulation can be described by a linear combination of shape basis functions. In this way, we can project the body curvature profile into the reduced shape space. We notice that the trajectory in the reduced shape space is an ellipse here, which we fix them by an oriented ellipse. To test the accuracy of the fitting, we compare to reconstruct body curvature from the fitting, and it seems like this reconstruct have closely reconstruct the original body curvature profile and the body postures. A perfect traveling wave can be represented by a circle in the reduced shape space. As you can see that for snakes, it's almost a perfect circle. And a perfect standing wave is a flattened ellipse in the reduced shape space, which is closely the fully limbed lizard you see here. We observe that as the leg size reduces, the wave of body undulation transits from a standing wave to a traveling wave. So here we show the quantitative data. It indicates that the transition from standing wave to traveling wave is then related to the degree of body elongation and limb reduction. But how should we understand such transition from a standing wave to traveling wave? What's the benefit of such transition? To answer these questions, we use geometric mechanics framework. Geometric mechanics is originally used to study the swimming at low Reynolds number. The idea is to decompose the configuration space into a shape space and a position space. In this way, we can interpret locomotion as a sequence of internal shape changes that leads to a displacement in the world frame. Let's start with a simple example, the swimmer. Its internal shape can be uniquely determined by joint angles. We call those joint angles the shape variables. And the space formed by shape variables are therefore called shape space. Assuming the quasi-static motion, the environmental interaction can be embedded into the shape space as a vector field, such that the displacement can be evaluated by the line integral along the vector field in the shape space. According to Stokes' theorem, the line integral is equivalent to the area integral over the curl of the original vector field. We call the curl of the vector field as a height function. In this way, the locomotion performance can be intuitively analyzed and optimized by the height functions. Geometric mechanics can be applied to systems beyond no Reynolds number. In fact, it can be applied to systems where the frictional forces dominate over the inertial forces. The ground reaction forces can be then measured and analyzed empirically. In previous work, it is shown that using geometric mechanics, we can design gates for terrestrial swimming. And here's an example here. So with all those analyses, it allows us to investigate the body movement in intermediate lizards. So from the previous analysis, we know that the body movements can be described in two-dimensional reduced shape space. And we coupled the leg movement to body undulation using the empirical body-leg coordination patterns. Here, we compared the body postures for standing wave and a traveling wave and some intermediate waves. And this is a height function for an intermediate lizard. So interestingly, two stripes emerge, which can be many useful gates. So the blue gates you see here, and the yellow gates here. So those are the essentially standing wave bounding one of the stripes. And the green gate is then a traveling wave. And interestingly, we notice that the traveling wave can enclose more surface integral than the standing wave. To test those hypotheses, we compare some simulations. So neither of the standing wave can yield effective locomotion. And there's an interesting gate, it's called a four-leaf clover gate. It's of particular interest because it is hybrid of two standing waves. Despite it's not being very intuitive, it is an effective gate. The traveling wave then outperforms the standing waves and has comparable speed as those in four-leaf clover. Thus, our analysis shows that the benefit of using traveling wave in intermediate lizards is to better coordinate with the legs. But what makes the traveling wave adventurous in intermediate lizards? So while it is a complicated question for biological systems, we can always start with a simpler locomotion system, which is a robot. So to better understand the theory of body-leg coordination, we built a robot. It can be viewed as a swinging swimmer with 4 legs, or can be viewed as a quadrupedal robot with 2 body bending joints. What is so special with this robot? So this robot both the leg and body can generate thrust. How should we balance and coordinate between the body and the leg? It becomes an essential question in designing and control this robot. So answering these questions can help us better understand the more complicated biological questions. In this robot, the body bending configuration can be represented by 2-dimensional shape space. Some of the regions in the shape space is not visible because there can be self-collision. Similarly, we can have standing wave and the traveling wave in the robot. And here we show 2 examples of traveling wave and standing wave here. So in our design, we first test the condition where the belly plates can generate thrust together with the leg plate. You see that there's a belly thrust place to generate thrust. And from the oblique video in the lower left corner, we notice that the belly thrust is contributing to the locomotion. Interestingly, we notice that the traveling wave can be beneficial when there is belly thrust. You see that on the left-hand side video, this robot is using a traveling wave, and on the right-hand side, it is a standing wave. And the traveling wave has like higher speed than those in the on the right-hand side, it's a standing wave. So similar to before, we use height functions to explain our observations. The vector field and height functions are shown here. the traveling wave body oscillation can better coordinate with robot with belly strut because the negative volumes are distributed widely around the diagonal corners. So in this way, imagine a flattened ellipse and a circular gate. Obviously, the circular gate can achieve a higher surface integral can be achieved for ellipse with higher sigma. Then we test the robot without the belly plate. As you can see here, we remove the belly plate that can generate thrust, and there's an oblique angle that we can see that there's no belly plate to generate thrust. And it seems like there's no difference between the standing wave and the traveling wave from those two videos. On the left-hand side is implementing traveling wave, and the right-hand side is implementing a standing wave. Again, we use geometric mechanics height function to explain our observations. So from the structural height function, we observe that the standing wave body oscillation can sufficiently coordinate with a robot without belly thrust, since most of the negative volumes are distributed along the narrow diagonal line, which can be sufficiently bounded by a flattened ellipse. The thin distribution height function can be a result of the vector field being almost curve-free, except those on the diagonal line. So here we show the quantitative results. So from the results, we have the hypothesis that the traveling wave body oscillation is advantageous in the system where the belly can generate thrust. So given those hypotheses, we wish to test whether or not this hypothesis is true or in the biological systems. Given the importance of belly thrust, can we induce the use of traveling waves in the fully limbed lizards by manipulating the substrate? So here we have our system, we have some air blowing out upwards to manipulate the environment. We hopefully that the legs can no longer fully support the body weight, such as some of the forces are just distributed along the ventral side of the lizard body. So here we have the a fully limbed lizard moving on granular media. There's no bubble, there's no airflow yet. And here's UMA on the on granular media, there's no bubble yet. And here, so here's like we manipulate the substrate, and you see that the body undulation profile is slightly different from it is before, where it's moving on granular media. And it's more obvious on UMA. As you see here, there's the feature of traveling wave emerged when the lizards moving on the fluidized bed, aerated bed. So here is a quantitative data. You see that the node is propagating when the lizards moving in aerated field. It's apparent from this body curvature profile. And here we compare the aerated trials with the trials on the granular media, and a significant higher sigma is observed. So to summarize, so what is locomotion evolutionary pathways? So locomotion evolutionary pathway can be connected to body elongation and limb reduction. So specifically, once you have small legs, the small legs can generate less thrust, which facilitates the use of traveling wave. On the other hand, the body elongation, which can enable more complicated body locomotion patterns such as traveling wave. So it's interconnected process of body elongation and the transition from standing wave to traveling wave and the limb reduction. And why do we need to study the locomotion evolutionary pathways? Because we have this insights, mechanical insights can help us understand the neuromechanical developments and the evolution. And also in the robotics point of view, those insights can help us better controlling the robot traversing the complex terrains. So that will be the end of my talk. Thank you.
Thank you, Bashi. I don't see any question. Yeah, so Josh has a question.
Hi, Bashi. How are you? So the geometric mechanics thing is basically like a low Reynolds number fluids formulation for Newtonian fluids. And I was struck by watching the video you showed just a few minutes ago in the sand pile, where the waves don't really propagate out. The waves move a little bit on the scale of about half the animal's body length, and then they sort of stop as the animal was sort of generating waves, which does not seem like Newtonian fluid. So I guess my question is where does this approximation of being in a Newtonian fluid kind of break down, and how would you have to correct what you did for that?
Well, yeah, so let me go back to the slides. Let me think. I think it is the slides here that you are referring to, right? So of course the force profile, how the velocity and the ground reaction forces or interaction are different in low Reynolds number fluid and those on the granular media. So we won't say that it's exactly the same for the Newtonian fluid or on the other side on the granular media. So it's a similar stuff, but it's with different. You see that they have different drag anisotropy and they have different material mechanical properties, such that in a short words, it is the ground reaction forces formula are different. Yep. And those differences can those differences are making some of these snakes are the terrestrial swimming and aquatic swimming different.
Okay, I will quickly follow up like a hypothetical question, and then we will move on to Sihan's question. So if you imagine the same analogy in, let's say, in water, like for fishes with fins versus, let's say, an eel or something, right? So, I mean, how do you visualize that when you are actually surrounded by the entire media and it's not just, you know, even not just embedded in a granular media, so to speak, but you're like isotropically surrounded?
Yeah. So our locomotors are quite different from those of eels, because our assumption is like the inertial forces are significantly less than the frictional forces, such that there's no gliding. So once the locomotor stops to self-deform, so they do not glide, they just stop the motion immediately. So that's basically our assumption. So the whole scheme is quite different from those of the eel swimming in aquatic environments. So yeah.
Okay, so let's move on to Sihan's question. Sihan, please.
Great talk. I want to ask about when you analyze these travel waves and standing waves of snakes, you only used one wavelength. And how do you find this wavelength? Because I'm imagining there can be many different wavelengths.
Right. That's a very interesting question. So let me go to the slide here. That's the wavelength is different from different species. That's not, although that's not the focus of this talk, but you see that the lambda, which is the wavelength, is different, and it decreases as the L decreases. So basically that, and for our principal component analysis, it can be shown clear that the dominant wavelength, there's only one, and only two principal components, which is one wavelength, have the same wavelength. So those can capture over the 80% of the variance.
And have you analyzed how the interplay between different wavelengths affects the motion? Because I know that in bacterial flagella, these interplay between different wavelengths can be very important.
Right. So this is our hypothesis, like saying that in the forward motion, only one of the wavelengths dominates over the others, so such that the other wavelengths can be negligible. But in other behaviors, such as turning or like probing some others, so in those environments, so there's interplay between different for example, the turning can be reconstructed by superposition of two traveling waves, so and EDC, amplitude modulation and the phase modulation that makes them possible. So yes, so we also studied the C. elegans turning and omega term, and similarly we use geometry mechanics and reveals that the omega term can be reconstructed by superposition of two traveling waves, whereas in this case, because only one of the wavelets dominance over the others, so we just focus on this one.
Okay, thank you.
Marianne has a question.
Yeah, I was just intrigued because you said at the beginning that snakes do better in cluttered environments. So if you change the granularity of your medium for the robot, for example, do you expect that the one with the middle thing is going to perform better?
Let me see. So what do you mean by increase or decrease the granular?
Yeah, because I think if you have like larger stones, smaller stones, like the sand that the robot walks on.
Yeah, that's a very interesting question. Yes. So it's actually the key parameter here would be the drag anisotropy. So in only our conclusion is made based on the assumption that the robot is moving on the granular media where there is drag anisotropy. Well, if the robot is locomoting on, for example, the hot ground where there's isotropic environment, the conclusion would be different. And yeah, so basically, so that would be similar to our observations here, that there's no, because the belly can no longer provide thrust because it's no longer anisotropic. So it's only providing the drags. So it's essentially detrimental to locomotion. So it's more similar to the case here.
Okay, thank you.
Josh, do you have another question?
Sorry, I just didn't put my hand up. No.
Oh, okay, all right. Okay, so I'll quickly ask a question and then we will head towards the conclusion of the session. So in your initial robot, the one you showed at the very first, which I think mimics the house lizards that we probably see more often, it looked like that the tail is kind of decoupled from the rest of the body. So, I mean, so that you cannot probably tell the entire thing is actually propagating as a, I mean, not as a wave, but you know, where the limbs were more fully formed, the motion of the tail looked like that was kind of decoupled. So, but I don't, you obviously didn't use that as any of your models, but what would be your insight on, you know, modeling those?
I am a bit lost about this question. So
okay, so when you say that your lizard is actually making a standing wave, right?
Yeah.
In that case, the tail in those lizards looked like they could move more independently. And probably when you took those nodes, one at the abdomen and one probably near the thorax, you didn't take into account the motion of the tail. I'm wondering, is that the case? Or?
Right, yeah. So that's definitely an interesting question. So the tail, so for now, we only assume that the tail is an extension of the body movements, and we haven't thoroughly studied the use of tail in those lizards locomotion and also the skink locomotion. But that's definitely our future work. But one of the interesting observation or our preliminary analysis is that there is a optimal tail size that maximize the forward speed depending on different substrates. So that's an interesting future directions. And also for those house lizards or like some other common lizards. So many of the lizards, the tail are lifted or like they do not directly contributing to the locomotion. They more serves as a stabilization process. So that's at least what we believe could be the cases for the lizard tail.
Okay. Perfect. Thanks for that excellent talk and thanks for all the speakers. We will quickly move on to Bill for concluding remarks.
Well, since it's the first time we've done it, I wanted to extend special thanks to all of our speakers. Different format, different challenges, and I think everybody really rose to them well. Not the least of which is that, you know, it's just a few minutes before 4:30, so somehow we managed to actually fit everything in. I realize that's not the most important thing, but it does help for people who are, all those of you who are hanging in until the end. It's nice that end on time. It was really a great set of talks across a huge range of topics. And and great questions too. And as I said, what we learned in doing this is that there are vastly more interesting people out there than we have had space to hear from yet. And so we're going to do it again. So please stay tuned. When our solicitations come out, let's be sure that we see as many people as possible. And then let me also add that on November 12th, we will return to our usual format of 3 longer talks. On that day, it will be about genes, geometry, and development, which promises to be very exciting as well, actually picking up on a few of the things that you heard about today. So thank you all very much, especially those of you who've made it to the end, and special thanks to all of our speakers. It was a great pleasure.